अभी तो आ रहे हैं बच्चे। वालेकुम अस्सलाम रहमतुल्लाह। वालेकुम अस्सलाम रहमतुल्लाह। ऑलमोस्ट सब है, मैं इसको बंद कर दूँ तो शुरू कर। Okay, Assalamu alaikum. Can we start, everybody? Morning. I think today his strength is less. I think maybe yesterday's dinner was very heavy. It looks like. Okay. Okay, because I have to leave soon, so I have to start. I have a couple of slides, maybe 10, 15 slides. I want to give you some thoughts. So, for last 4 days, we have gone through what is the drug discovery and how we connect as an academic institution with the industry. That will be few thoughts. I will give you some reading material. You can get it from them, and these are not the drug discovery reading material, but science fiction kind of thing. But it is true, and I will explain to you, inshallah. So these are the 3 books which I have read. Okay, so one is Drug Hunter. I think if you can get in your library here, that will be really nice. So this is written by दो जर्मन साइंटिस्ट ने लिखा है इसको, और कैसे जो है ड्रग डेवलप की जाती है, ढूंढी जाती है, और उसके बाद मार्केट की जाती है. दिस इज़ ऑल, एवरीथिंग इज़ देयर. This was written by from 2 German scientists from Schering AG. Second book is very famous, The $1 Billion Molecule. Okay, and this was written by this was actually written by a regular writer, Barry Rath, but this is the story of Dr. Joshua Boger, who was working at Merck, and then he went out and made a company called Vertex, which is like about $80–90 billion company. So this is the life story of Dr. Joshua Boger. And how many people know about Venki Ramakrishnan? Anybody knows about Dr. Saab? Yeah, see, he knows. So, Nobel laureate. So, yeah, and the biology problem. So, gene machine. Venki Ramakrishnan is a senior scientist in Oxford, and he still writes. He don't have a car. He still rides his bike. So he's so famous. Very nice book. And you really enjoy reading at least 2 of them. Third one, if you don't want, I think you can ask your library to get all these books. Okay. So this will give you a glimpse from academia to industry, because Venki is working in academia, and he has done a lot of work on ribosomes, which is a organelle in the cell. And $1 Billion Drug is, $1 Billion Molecule is a industry book, and the first one is both industry and academic. So try that sometime, okay? So what we talked all along, last 4 days, right? We talked about the process of drug discovery, and then how it is going in academia, and what are the important part of discovery development. So we learned target discovery. We have given a lecture. Then hit generation, lead generation, and this is called, we call it exploratory. Just I'm capping up so that you understand what we talked. And then candidate, we select a candidate, we go to NL model. Dr. Zainuddin talked about all those things. Lead optimization, I talked in my own area. Then clinical trials, we have given several examples of clinical trials. And then development, and then it goes to the approval and then post-approval. So this is the complete process of drug discovery. If you want to read more, there is a book there, okay, down, Deline et al. In book Introduction to Biotech Entrepreneurship from Idea to Business. So you are going to have idea. We discussed yesterday a lot of ideas. If you want to know how to go to business, read this book. Okay? And Dr. Syed was here. He came from a lab to entrepreneur. He has about 300 crore business now. So that I'm sure that he read this book also. Okay? Okay, so there was I have 5-6 more slides, and this was I wanted to tell yesterday, but we went into different mode. So I want to show these slides that how we develop a project. Okay, you guys were developing a project, so it was good that I did not show it yesterday. So now you think about how you are going to develop a project. This is all my work, and Dr. Zain used the word serendipity. Okay, so this is all about serendipity, and we are very, very successful in developing some molecule which are really good. Let's go quickly. Introduction. Serendipity. What is the meaning of serendipity? It is happy accident or accidental discovery, you can call it. Refers to chance discovery. Making discoveries by accident and sagacity of things which are not you are looking for. You just see that it happens, okay? It is like that. And this was coined by Horace Walpole, 1754. The modern day therapeutic discovery is serendipitous drug, including empirical discoveries. It is not always unsought. So serendipity is a built-in drug discovery. And if you see the next slide, how many drugs are serendipitous? You can see that once you come up. So I can explain. This is a little bit older slide. It is about the same. Now, 2025 also, it is about 1450 drugs. Because I took this from this paper, okay? So that's why I'm putting there, but I know exactly. So about 437 drug molecules are on market, and approximately 24 have some element of serendipity, chance discovery, Somebody was making something, mixing something, and he got something, okay? So it became a drug. So this is called. The pie chart shows that how many there are, 75% is not serendipitous, and then others are. In case of cancer, it is more. That's what, because we talked a lot about cancer, okay? So in cancer, it is more, so there are 64% are non-serendipitous, and there are 14% are serendipitous, and the derivative of serendipitous are there. So 88 drug molecules on market, 13 are serendipitous, 18 are chemical derivative of those 13. So there are about 35 clinically used cancer drugs. So, we call it drug by chance or drug by luck. And we chemists always say, as a medicinal chemist, that we have to be lucky also. So for people like you are looking for Nigella, right? Sativa, you have to be lucky. So you need to pray a little bit, okay? So we do that, okay? So it happens. Okay, so example of serendipity, I think one of them was given, but I thought that I'll just give you a couple of examples. Penicillin was discovered in 1928, discovered by Sir Alexander Fleming, and it used to kill the bacteria, antibiotic or bacteria killer. And when he saw the mold on the plate, he said, it looks funny. That was his word, it looks funny, okay? Then there is a sildenafil citrate, this is a lifestyle drug. And then discovery of Lytic, which is used for the eyelashes, okay? Increase the eyelashes, and that was also discovered by chance. There is a book about that, again, you can read that, and I met fortunately this person. and he has given a talk in our institute, Martin. So you can look at how, there's a whole list of serendipitous book there, a drug there. So you can look at that. So this is also part of our game, that we look for serendipitous research. Now you will say that, oh, I'm talking about an old timer. No, I will give you a very new example. This is JAK inhibitor, COVID-19 inhibitor, baricitinib. This was discovered by chance also, okay? And in my family also, my sister had COVID, my sister-in-law had COVID, my father-in-law had COVID. Two of them at that time we get somehow from the Baroda, but my father-in-law could not get, so he passed away. So this is a drug which was meant for arthritis, okay? But it was used, so it is written all there. Early in the pandemic, there's a company called Benevolt, so AI we talked about a lot. So they made this chart, and they found out that this may work. Again, I am using word may work in that. So this is the pathway, and it is an anti-inflammatory drug, so it worked at that time, okay? So this is the new serendipitous drug. Okay, what I have done, because I'm quite famous in our institute that I really I'm a believer in serendipity. So I thought that 4 or 5 slides I quickly do that. So I have a collaborator. His name is Professor Ravi Al-Kaisi. And when I came 10 years ago, and he was making photochemically responsive crystals, he makes some crystals which are energized, and he used to publish a lot of things. And I told him that, why you are doing that? Let's work on some anti-cancer compound or some anti-tumor compound or anti-ulcer compound. He said, no, I don't want to get into that. My area is focused. I just want to do that. I have done my PhD, postdoc, 2 postdoc. I don't want to go there. So somehow I convinced him and got some compound from him and got it tested, because we have Dr. Aziz was also there. So we tested MTT, we call it MTT assay. It became a full project. I was funded about 1.2 million real for that, and I will show you how we got there. 5 more slides. Because it was long presentation, so I cut it short so that you can understand. So I told Dr. Kaisi that you are making some compound, which you say that it will not be a drug, but look at this paper. When I showed him, this is the compound on your right is explosive. It is used in satellites and other sending satellite in the. So I said this compound was done by this compound was synthesized by some people, and they are doing clinical trial. It is in phase 3 clinical trial, and it was from energetic compound to very nice anti-cancer compound. And when I showed him paper, he was still not convinced. He said, no, I'm not doing anything drug discovery. No, I'm not going to do any drug discovery. So it was tough for me to convince him to get some drug from him. People are very. So when I showed this paper, then he was a little bit soft. I said, okay, I'll give you some compound. So that's what I wrote, that we developed a new class of anti-neoplastic agent from photocally responsive crystal to anti-cancer compound.
Academicians love to see
Publication, yes. And from me, he got first patent, which I'm going to show you, and he is very happy.
That's a different level.
Yeah. So anthracene-based photochemical responsive compound, so this was the preliminary result. I will not read everything. Just I want to, these compounds cannot be a drug, okay? These compounds, they are very greasy compound. They will not cross blood-brain barrier. They will not cross, cell membranes. So when I saw that, I said, give me, Professor Kaisi, couple of compounds. Let me test. So we tested some compound. I'm not showing that slide. And it was active. A good amount of activity we got on. Then we keep on, optimizing. You hear the word optimization a lot. So we keep on optimizing this molecule, and we got two surprises, which you'll see on next slide, and they all are patented. So what we did now, from below there was a 3 ring, okay, those who are not chemists, that was 3 rings. So we changed, I designed molecules, we changed the rings, we changed the upper part also. So if you see the compound on your left, first compound, that was in MDA 237, this is a triple based cancer cell line, and HCT is, remember, HCT is a colon cancer cell line. So it became 1.37 micromolar. We changed the nitrogen, so this is optimization. That's what I want to cap up, the optimization part, that how chemists think. When we switch the nitrogen from there to there and switch the connection, you can see that MDA 237 is 1.375 and HCT is 12.3. So we got an element of selectivity within cancer. Number one, we got a compound which is equivalent to mitoxantrone, which is already marketed drug. So my objective was complete. I can close the project now because that was the objective. But no, we went further. We got this compound, and this was, we call it dances with nitrogen. So this was like between me and him. He was very excited, and then he said, I will change the upper part, and he did many things, and we got a very good compound. Yeah. So we did not change any nitrogen because we got enough nitrogen there. Solubility was not a problem, but nobody used the upper part. That was in literature, we could not find anything. So we got it patented also. And this was, I think, the first patent. Yeah, one more story. So when we are doing this work, we saw a paper coming from from the UPenn, University of Pennsylvania, very close to our work. I said, who stole our work, right? So look at this molecule, and I will again click one more time, then you will understand better. So I just made these molecules. Look at that. These molecules, phenyl, thiophenyl, naphthylfuranil, but it does not have the nanthracene. So that's okay. We are not going to go for anthracene. We made something else. We changed the upper part to the what I showed in previous, which is called 1,4-dioxylane. So we immediately went into action, and we immediately went into action, and we patented our molecule, and this became first-in-class dioxylane compound. We got the US patent, we got the Saudi patent also. We were very fast, we confirmed, and this later we did some mechanistic study, and we found it is a topoisomerase inhibitor, which is a target. I think people were talking about target yesterday, so we did some work. It is very specific for breast and colon cancer, easy scale up. We patented, so we have a robust protection. And we talked to some company, and hopefully they will be licensed. But we don't have the animal data, so we are going to get the animal data soon. Okay, everyone was talking about the technology and AI and this and that. So this is, I'm just trying to convince you that technology is good to use, but sometime you have to think about this graph, okay? Technology goes right up, and we are very happy, then suddenly it does not work, then it comes down, then it becomes plateau, like that. That is where we should work. I will leave this slide with you. You can read all those things, and there is a reference there also, okay? So technology, it is good to use all the time, but it is not always that, oh, AI is going to solve the problem. Dr. GIAN was very nicely, he was telling all the time that I want to see a drug by AI. You have to take this graph with you, okay? This is what it says, hype. Go on the people. What we call it, dhadam, right? And then it will plateau. I'm not saying don't use AI. I'm not saying don't use, but be very, very careful, okay? You have to put your all effort into that, but think about outside the box also. So I'll leave this hype cycle with you. There are minute things are written there. I think it is worth reading for everybody, okay? So this is the, I think, the last slide, I think. And my friend, because I always tell about Professor Casey, how he became a believer in drug discovery before he was not a believer in drug discovery. And now we meet every day, 2:30, we drink coffee together, and he is the one who writes on anywhere, anything, you know, on paper, on glass, in bathroom, on tissue paper, everywhere he writes. And he is working with me, and he's doing his work, and he's discovering more drug. And he has a very, very nice paper and patent who is going to publish in JACS. So JACS, yeah. We already published one with him, and we are going to working on the publishing another paper in JACS. So he knows more medicinal chemistry than me. He knows more kinetics than me. He knows more enzymology than me, because in 10 years, he read a lot. So take home message for you, read a lot, okay? This guy was not ready to do anything other than what he is doing. Very stubborn. He's an Arab, so it is very difficult to convince him. But I convinced him to do this, and now he is with me, and he is moving to actually University of California, Riverside soon. And this is me and him. This is an iconic picture. We are standing in a conference, and he was having coffee cup, and then he said, "Oh, can you tell me about this?" And this photo was circulated all over, you know, in Saudi Arabia, looks like. So we call it iconic paper, that he can write on anything, chemistry. So that's what. So I thought that this is my final slide, and I wish you all the luck. Thank you for getting me here, to organizers, to Dr. Azhar, and meeting all of you. Whatever I know, I could impart, give you, and wish you all the luck for your future endeavor, and please be in touch. Thank you so much. Any questions? I'm still here. I'm not in the car. Okay. Anything you want to know more? I think that will be possible later, not today.
This is the time you can shoot your questions.
Yeah, this is the time you can shoot.
Around, but these two people, they will not be.
Uh-huh.
So please utilize.
I will be just an email away from you, WhatsApp away from you, okay? And I will really like to help and see some of the ideas which you discussed yesterday, how you connect with industry. I will say one more thing. You made a group. You know this group of the drug discovery scientists now. You are a drug discovery scientist now for me, okay? Because the way you presented yesterday was incredible, out of my mind, okay? So you make a group, this group, you make a you know, and work on 1 or 2 projects together, okay? Just write up something, send it to Dr. Azhar. Maybe he will send it to Dr. Sayyad or to me, and we can connect you with some people who are really good in assessing whether it will be viable or not. So this will be my idea to them.
That's an excellent idea.
Yeah.
Because we had 48 participants, right? And we can see that some of them, they were really, I can say some of them were not sure why they joined, but most of you who were interested in and you went along the whole course, we can work together at least to write something, right? Kind of a review or a meta-analysis or something. And Dr. Imad, Dr. Zan, we can be all together.
Yeah.
So shoot me your interests, that then we can work out a structure and go from there. Because I know in academia, whatever you say, even I have one slide, that whatever you say, publication is something we always crave for. There's a craving for publication. Everybody wants to publish, right? And it does help in academia.
No, we published also, but we patented also. I showed you both the thing, okay? Yeah.
So I think that is something that you can see coming out from this course, that you have got a tangible outcome, that you have a paper or publication. It doesn't matter, I mean, it could be several authors. Authors is not a limit, because now even I hope that more people would be contributing more, and it can increase the quality of the work.
It will be really nice idea if you sit down together, or few of you sit down together, think about the target, read the literature, write a review, and that
Yes,
will be,
I think,
yeah. I know you have not done any experimental work, but you can write a review without experimental work, and you can say that we learned from this course, so we are going to do that. That will be
Yeah, I
really
think
good. This
that's
will
an
be the
excellent
best
idea.
outcome of your effort here.
And who knows, that may be a primer for some future projects,
Yeah.
right? So while working on it, you might find some smaller subgroups who can work together and have some projects for your masters or your PhDs or postdocs or whatever.
I remember there were topics about neuroscience. There were topic about CDK1 kinase. There were topic about Nigella sativa, and then there were other topics. So, you know, if you think, and it is not necessary that you stick to that. If you think something else, talk to experts here. Talk to us, and you can at least put a one-page summary that you want to do that. That is not a big job. Then we can work on it.
Yes, we can work on it.
We
That's an excellent
define it.
idea. I think I'll take it forward,
Yeah, inshallah.
and we
Yeah.
can include it in our recommendations as well.
Okay.
So what other things that I would say, you would say in academia while they are here, and if they want to move to an industry, what would be the do's and don'ts that they should be looking
So
for?
in industry, you will be bound by the university rules, okay? But never, you know, you can ask that, but you never give up. You can put your idea to the management or somebody. They want you to work for their specific job. That's why they are paying you. But you keep putting them, and you ask that, I'm working 90% time I will work on your work, but 10% time I want to have some idea and I want to discuss. I'm not doing anything, but this is what I want to do later. So tell to your supervisor. You have to be persistent, because initially you will hear no, and again you will hear no, but you go for 3-4 times, you will hear yes. Let's see what is there. Okay, there you should start doing that. So that's the.
Sir, now are you having some nanoparticle based drugs?
Mm-hmm.
Sir, any drug have you worked, sir, where you are using nanoparticles for that? So I don't know, sir, is the nanoparticle being used for the drug discovery?
Mm-hmm. Yeah, it is. There are nanoparticles already in use as a medication. Doxorubicin nanoparticle are being used. That's your question, that it will be used or not.
Has it been used already?
Yes, they are not one of them. There are many of them. Just Google it and you will find many nanoparticles are already there. Which one is there? Lipid nanoparticle is there, iron nanoparticle is there. That you have to figure out what you are going to do.
Actually, when I was in an MTech nanotechnology, so our sir, he said that doxorubicin, when it is capped with certain anisotropic nanoparticles, which are not circulating safe, so they have absorption spectra in both, and ani region also, and absorption peak in 2 region. So by that, what we can do, we can target drugs to the tumor region, and we can add radiation. With that, we can heat up that cells. So we can attack that cell from 2 ways. So is that anything that you have come out?
Everything is possible. I think you have to design in a way that if you are giving as an injection, you cannot inject anything in the tumor. It is not going to work that way, because pure tumor may be already spread out also. So you have to design nanoparticle in a way that first thing, like tumor, solid tumor especially, they are hypoxic. So you have to design something which reaches there, bind into that, and in hypoxic or oxygen-less atmosphere, it is cleaved. So that's
If I understand your question correctly, he's asking if there's any nanoparticle which is serving as a drug. Because usually nanoparticles have been utilized
It's a
as
carrier.
carriers.
It is a carrier,
That's
not as a
not
drug.
carrier.
Yeah.
I don't know about any nanoparticle that is used as a drug.
Actually,
Was that
nanoparticle.
your question?
Yes.
Yeah, not nanoparticle alone it is not a drug. It is a carrier. Actually, nanoparticle alone is a question. It may be toxic also.
Yes.
So I thought that you were asking that nanoparticle with drug. Nanoparticle is always used with drug or as a carrier of the drug.
That
Okay.
was also one aspect, sir. I'm saying, sir, with the drug, we are adding a receptor, we are adding a molecule by which we will target that tumor, and that we are adding that nanoparticle. For example, with doxorubicin, we are adding a lectin for receptor, or which we will target in the tumors, some receptors or some targets, and with that we are attaching the.
So you are just adding that. So
nanoparticle.
it is excipient, what you call it. Okay. Excipients are already built in the molecule or the cancer molecule. So I'm still trying to understand because see, nanoparticle contains a drug. It is encapsulated, it is on the surface, inside the surface, outside the surface. There are many things are there. But doxorubicin, like example, there is a nanoparticle based doxorubicin, which has a better half-life and it has a low toxicity. Why? Because it is encapsulated.
Modulating the drug.
Yeah. So you have to always.
Sir, is it possible as a bachelor student to write a project or enroll in a project?
Which period?
In a bachelor, pursuing a bachelor, is it possible to us to join any project?
Doctor.
I can take that question. See, in your bachelor's, it's mostly classes and projects, right? You can be involved, but it will be short term. I don't think you have a dedicated last semester that you can do a project. So you can utilize that part of your curriculum that allows you to do a dedicated project for 6 months.
There is no curriculum in our course. There is no research project
So
in our
prior
course.
to do your graduation first, so you have to follow the curriculum.
Yeah.
Not do a project and fail in your.
Don't fail there.
Sir, but if we have idea, some novel idea, and discuss with other, is it possible to join as a officially in a project?
As I said, your curriculum allows that. You can't miss the classes. You can't just do research and while your classes are going on, your attendance is short. Let us talk about it later on. It's an internal issue. How to take that out.
Anything else? Okay, thank you so much. And I have to. Anybody else? Okay. Okay. Good luck. My best wishes, and I will be just leaving it here, and I take my. I will leave this slide. There are some references here. So I think this one, or I'll send it. Okay, that's good.
I'm uploading everything on the cloud. Were you able to access it?
Yeah, I saw yesterday. My slides.
So some of the presentations are not there, but most of them are there.
Yeah.
And that will be enough for your exam.
For my only this one is not there. I saw yesterday.
Chromosome 6 was creating humanized bonobos that can utilize the rich table to get any organ if they need. Very interesting. Chromosome 6.
We used to watch Star Wars. Now it is like not anymore. It is a fiction.
US is already developing their space army.
Yeah.
They already have a ministry that is taking care of space wars. So ideas can always be
These
translated.
books are real books. They are written by real scientists about real scientists, and it is not the actually I read Dr. Ravi Machine and Mr. Maybe I read the book. I don't know which one. Okay. Assalamualaikum.
So
Thank you
we
so
hand
much.
over the podium to Dr. Zainuddin. Let's give him some time to relax.
Yeah.
The class is all yours. I will send all the contact details. I'll give a contact person for all units that you want. Yeah, we will work on. Let's see how many of you would be getting on board.
यहाँ से निकाल दिया, इसको निकाल के रख दिया, है उनका है ना, मुझे याद करके दे देना, उस दिन मैं भूल गया था, आज हाँ वो मिल गया था, मगर हमको आज फिर हम निकल जाएंगे ना हम लोग।
क्योंकि मैंने आपके लैपटॉप के पास बगल में रख दिया था इसको।
So, hey gentlemen, if you don't mind, can you come forward, please? Yeah, come sit. I will tell you some stories today. Huh? Mm-hmm. Wow, it's been 4 days. Today is the 5th day, probably. It's been exhausting, exhilarating, and probably it will be good for you guys to know something more, something a new science, you know. And we all learned together, and we also learned that there is a spark in the people who have attended. They need something to understand. With no doubt that it's not important that you guys all should understand everything, no need. Just to get a leaf out of it, that will be one of the thing. At least it will help you out to make some advances in thought process. See, research is the thought process, nothing more than that. You have to think of it, that's all. And I told you on the very first day, as the Linus Pauling said, the best idea to have a lot of ideas. So be a thinker and go with the small bits of a thing, and I can tell you, you all will succeed once again. Yeah, I have to take this call here. So today we have a topic. Let me talk of a framework for academia-based drug discovery. We all have seen that how it is, and we all spoken a lot on it. So I start with the saying that future is bright and collaboration is the only way we can do it. From industrial perspective, of course, this is a collaboration which we have to do. And the mantra for it is forming a team, working as a team to target the devil. And the devil can be a disease which we are talking. It can be a cancer. It can be any of the disease which you are working. So it's important to combat something from many sides. That's all. And you guys, see, best part for you guys are, you are not a contaminated brain, you know. You guys have a fresh mind, and think of it. Thinking is a limit. Think of it, how you can combat. For that, only thing is your basics should be good. So make sure you are brushing up your basics nicely. And that's the important part. If you are a good molecular biologist, read it nicely. If you are a cell biologist, you read it. If you are a signaling transaction person, read it. I'm telling, make sure your basics for everything should be good enough. Then it's easier to, there are 3 levels, you know, of teaching which you say. One is the basic level, another is the informational level, and third is the philosophical level. So I need everyone to have a basic first. Good level of a basic helps you to put a lot of information in it. And there is a lot of information floating across. Then I want to give you all that happy news that cancer drug, I was just giving an example only for cancer. See, these are all drugs which are billion-dollar drug. It's all came from academia. So you guys are playing an important role. Don't say that you are led behind or industry people are led behind, taking a lead. No, you originate, you partner with us, and then we take it forward. So it's a seeds come from academia, and that seed becomes a fruit-bearing tree, and that is what we use to do it. Okay, so it's more of a collaboration and academia industry relationship. That's the most important part. Next purpose is what is the purpose for academia, India, and industry interface. So important is interface with the industry, with the transfer of a knowledge and technology solution within the research center. We all explained from last 4 days, we are all keep on repeatedly talking on it and saying that there is a technology. See, don't restrict to your anything. Sir was telling just now that there is a molecule, you can make a molecule, think of it, make it a small molecule or a big, large molecule. On a chemistry wise, you can synthesize it. From biology wise, you can think a process, or reading your book and all, and say, oh, these guys have target, Google it out and find, oh, RAF is there, is it any drug which is hitting that? What is the reason why it has not been hit? Think of it, and hopefully you will get something. So any idea, novel which is, or which has its own way, discuss with the expert and think of it, and you write a project. Be a pharaoh. I just want to tell you guys to read a lot. And why everybody says, in my MSc's days I used to read a lot of fictions, you know, and still I used to write Russian. Isaac Asimov was one of the fiction writer. We read a lot, you know. He has a lot of fiction science books, you know. So what it does, it tells you how to increase your thought process. See, something is beyond which is not realistic, that becomes a fiction, and after fiction becomes a reality after some time. Okay? So be a reader. Get your life out of these mobiles, you know, and focus on reading. Mobile is also a mean to read it, okay? But read a lot. I think contributing to the entrepreneurial or entrepreneurial competencies, contribute to value creation, impact university for the several innovation channels, and that's how people now, you know, I will be talking more on it, that there is a fund crunch, but people are there to give you money, provided they see. I was here listening one thought from a Nobel laureate, and he said, your research is as good as the $1. What does it mean? It means if you have something, if you go to the market, if somebody pays you $1, it's a worth. Okay. Research publication, it's all okay. For academic means you need. If you would have not published, other people would have not read it and they would have not climbed to that position. So publication is important, knowledge sharing is important, but do something which can be worth in many ways. So I am just giving, these are the, this is a central dogma for drug discovery. We have repeated this slide many times. Target identification, hit discovery, lead. So I am just giving where is the role from academic and where is the industry role. Academic, industry, they all go parallel to the preclinical. So these are the zone which tells you that here you can, these are the zone where you can tell that you can work from academic perspective. Small things, he was telling you make a small molecule, you do a assay, you do small things. I am telling you do one assay, you catch up with the lab and say what you do, can you help me out. When I was doing PhD here in AMU, we were doing on something a tocotrienol rich fraction, wanted to see that CHD, whether the chronic heart disease and all those things, and then we were working on a rabbit. So we dealt with some of the people with the anatomy department and other department. They took out the whole aorta and then they scrap it. I didn't did it, they helped me. So there was a cross-functional work, and we were working with Dr. Jamal also. We published a couple of papers with those things. So what I'm telling, at that time we worked together. We worked with 3-4 labs, department, and then only my thesis came out, you know. So I think you all should know which lab is doing what. That is more important. Know-how is important. I think that's the best part. And which lab has instrument. I don't know how quickly you can access those instruments, but at least the PhD student who will be working will help you something. If you want to get a molecule, you want to clean it up, you want to run HPLC or LCMS, ask those guys to run one injection and help you out with that. And then you should have some information. So this is the way science is. Nobody can do alone anything. So think of it, make a procedure. That was what we were doing. You all got an idea, but first thing is an idea. Second is how you will pursue with that idea. And third, how realistic that idea is. When you start writing how you will pursue step by step, when you say step by step, I have to go out of the university, I have to write, I have to take 5 steps here, move right side, open the door, root outside, there will be a couple of people standing and they will ask, hey, where are you, why you are going inside? I have to answer, move out, get in my car, open it, switch the switch. You have to write small. That is known. What is the difference between protocol and SOPs? Anybody knows? Anybody knows what is the difference between a protocol and SOPs? Yeah, both. Okay. Protocol give you an idea. It's still, you refine, you say how you have to do all those things, detail. But the detail, as standard operating procedure is known as SOPs. SOPs is one I was making in my lab SOP around 20 years back in industry. There was when we used to make SOP, the concept was if I give my SOP to someone and if I bring a college boy of 12th standard or a 10th standard, he can perform that assay. That is known as SOP. You don't need knowledge, you just read it, it will tell ट्यूब उठाओ, रखो, पाइपेट उठाओ, उसके अंदर टिप लगाओ, इतने ml की पाइपेट है, 3 ml की, 1 ml की, 0.1 ml की, वो उठाओ, उसको रखो, उसको क्लिक करो, उसमें लगाओ, फिर बफर डालो, बफर वहाँ रखो, वहाँ पे टेबल, मतलब उसमें ये है कि आप एक एक स्टेप आपके पास लिखा हुआ है, तो दैट इज नोन एज एसओपी। तो जब आप भी प्रोग्राम बनाओ, कोई प्रोजेक्ट लिखो, तो एक एसओपी बना के डाल दो उसमें। और एसओपी एटर्नल होती है, हर लैब की एसओपी एटर्नल होती है, उसमें कभी कोई चेंज नहीं होता, वो चिपका दिया जाता है कि जिंदगी भर कोई आता रहेगा, आपका नाम लिखा है और चलती रहेगी एसओपी। खैर, तो ये है इंडस्ट्री कहाँ रोल प्ले कर सकती है, एकेडमिया बहुत जगह दोनों साथ काम कर सकते हैं। अब चैलेंजेस क्या है रिसर्च की आजकल यूनिवर्सिटी में, जो मैं समझता हूँ बाहर से। फर्स्ट इज लैक ऑफ सपोर्टिव इकोसिस्टम। ऐसा लगता है जहाँ ये कह के हम ख़त्म नहीं कर सकते हैं, वी कांट ब्लेम एंड मूव फॉरवर्ड, नो, वी हैव टू सॉल्व दैट, ओके, एंड दैट्स onus is on us, okay. There will be hindrance, but there is onus on us how we can solve this problem and how we will make this ecosystem a better. हमारे पहले वालों ने जो किया उसकी गलती हम नहीं करेंगे, हम आगे एक अच्छा ecosystem, at least this group, you all a group meet together, have some group discussion, we learned it. I am a physicist, I am a mathematician, I am agriculture sciences, I am a nanotechnologist, I am a biochemist, I am a molecular biologist, I am a molecular medicine person, I have a versatile group here. Why don't we do one thing? Why don't we make a small cross-functional team? Let's float an idea and say, this is an idea, what are the things which you will be filling in these boxes, and can we write a proposal or a review at least and publish a paper? No paper is good. People love to have one name paper. No, I have a lot of paper, and we have people of 20, 25, many people from Harvard, people from many different places. I can show you some paper which we publish, and that is how it is. More the people, more the beautiful paper you can write it. डोंट बिलीव दैट मेरा नाम हो जाए, मैं लिख दूं, ना ना ना, पेपर अच्छा होना चाहिए, उसकी साइटेशन वैल्यू करती है, दैट इज मोर इंपॉर्टेंट, और कितना इम्पैक्ट फैक्टर मिलता है वो इम्पॉर्टेंट है। एक पेपर से 8 अगर स्कोर जाओगे तो इतना ही मिलेगा। कुछ पेपर हैं जो 15-16 साइंटिफिक रिपोर्ट्स के पेपर है, साइटेशन बढ़ती जा रही क्योंकि वो रिलेवेंट पेपर है, ठीक है ना। तो ये कोशिश करो कि ये ना करो कि ये तो ये ग्रुप है, इस पर एक तुम लोग पायलट प्रोजेक्ट डाल दो, एक ग्रुप को मेंटेन रखना अपने पास, इस ग्रुप को, एंड थ्रो आउट द आइडियाज एंड से लेट्स मीट ऑन अ कॉफी, लेट्स डिस्कस दीज थिंग्स, यू नो, एंड यू कैन डू इट. Take help of Dr. Azhar and he will guide you once you write something preliminary on it. Okay? So communication should not stop. You paid it, you came it, you joined it, you left it, and then bye bye, we will see together once a year back something. Okay? Don't do that. Take this legacy forward. Positive funding balance, that's a challenge. Nurturing collaboration and entrepreneurship, that is one of the things people don't think. हर आदमी डिग्री लेना चाहता है और जाना चाहता है. मैं एक एग्जांपल दूंगा, अभी कल ही की न्यूज़ है, मैंने लिंक्डइन पे देख रहा था, और आई विल शो यू. मोस्ट ऑफ द बिजनेस डू नॉट हैव कॉन्फिडेंस, यूनिट्स इन द कंट्री डोंट हैव कॉन्फिडेंस. तो ये मैं बार बार बोलता हूँ, क्यों जो है हम लोग एज ए इंडस्ट्री कॉन्फिडेंस नहीं है. मटीरियल सप्लाइड बाय देम टू द रिसर्चर विल नॉट बी मिसयूज्ड एज सच, दे ऑफन रिलक्टेंट इन सप्लाइंग इन नीड ऑफ इंफॉर्मेशन टू द रिसर्चर. ये एक सबसे बड़ी चीज़ है एथिक्स की. हम ये बिलीव नहीं कर पाते हैं मैं अपना मटेरियल दूं और उसको वो हमारे ही काम में यूज़ करेंगे, कहीं ऐसा ना हो कि वो उसका कहीं मिसयूज़ कर लें या उसको यूज़ कर लें. उसका स्ट्रक्चर अगर हमने तो कोई नहीं देता है, मगर कुछ इलस्ट्रेट कर दो, इसलिए थोड़ा इनहिबिशन है. तो ये भी हमें ट्रस्ट पैदा करना पड़ेगा और बहुत इंपॉर्टेंट पार्ट है एक इंडस्ट्री के कोलैबोरेशन के लिए. देन इनएफिशिएंट इनसफिशिएंट इंटरेक्शन विद द यूनिवर्सिटी अक्सर होता ही है. अब हम आज आए हैं, पता नहीं 2-4 साल बाद कोई और इंडस्ट्री से आएगा, कोई बात करेगा, तो फ्रीक्वेंट रिलेशनशिप नहीं रहता है. लोग भी जो है अपनी ज़िंदगी में मस्त हो जाते हैं, एंड देन दे लीव, दे मूव फॉरवर्ड. तो दैट शुड नॉट बी देयर. देन यू हैव अ टाइमली सेक्रेटेरिएट असिस्टेंट इंक्लूडिंग कॉम्पिटेंस एंड ब्रॉड टू नैरो. ये सब फोकस बहुत है. What drug discovery industry is looking from academia? That's very important point. कि क्या चाह रहा है. हम ड्रग डिस्कवरी की इंडस्ट्री जो है एकेडमिया से क्या एक्सपेक्ट करता है. हम बहुत कुछ एक्सपेक्ट नहीं करते. हम चाहते हैं टारगेट आइडियाज़ आपकी तरफ से आए. आइडिया की कोई लिमिट नहीं है, इसलिए आइडिया एक ऐसी चीज़ है जो किसी से भी, पढ़े लिखे से भी मिल सकता है और नहीं पढ़े लिखे से भी मिल सकता है. वो भी सोच सकता है सर ऐसी बीमारी है, ऐसे इसका क्या कर रहे हो, कुछ तो करो इसके बारे में. वो कुछ और भी बोलता है तो आदमी का क्लिक कर जाता है. देयर आर लॉट ऑफ स्टोरीज लाइक दिस. पीपल वेंट टू द कैब ड्राइवर, टोल्ड समथिंग, पीपल वर सिटिंग इन द बैक सीट एंड दे थिंक ऑफ इट, दे वेंट टू द लैब एंड थिंग्स हैव चेंज एंड दे बिकेम समथिंग न्यू, दे केम अप, यू नो. सो दीज आर द स्टोरीज. कैटर नैनो टेक्नोलॉजी बेस्ड फॉर्मुलेशन, ये सबसे बड़ी चीज़ है. आजकल बहुत सारे ड्रग, आईवी दिए जाते हैं, ओरल नहीं दिए जा रहे हैं। ये कितनी अच्छी फील्ड है नैनो टेक्नोलॉजी के लिए, खासतौर से एब्जॉर्प्शन टारगेटेड डिलीवरी के बारे में। वहाँ अगर आपने कर दिया तो आई थिंक यू विल डू ए हाफ ऑफ इट जॉब। और जो नैनो टेक्नोलॉजी में एक्सप्लोर करो इसको, ये फील्ड बहुत बहुत ज़्यादा इम्पॉर्टेंट है। हर आदमी चाह रहा है किसी तरह से हम कोई फॉर्मुलेट कर लें, कुछ उसके अंदर कर दें ताकि हमारा ड्रग, क्योंकि तुम भी जानते हो कितने लोग हैं, अपने घर में जाओ और कहो कि ड्रग देना है तो ओरल दोगे या आईवी दोगे। हाँ ले ले, पेशेंट कंप्लायंस जो है वो ओरल से है, आईवी से नहीं होती है। तो जो आईवी ड्रग इसलिए मजबूरी में बने हैं, और कुछ ड्रग है जो ओरल भी है, आईवी ज़रूरत से भी बनते हैं, मगर जो मजबूरी से बन गए, जिनकी सॉल्युबिलिटी नहीं थी, एब्जॉर्प्शन नहीं था, उनको बहुत प्रॉब्लम हुई, तो वो लोग नहीं बना पाए। तो इसलिए नैनो टेक्नोलॉजी एक बहुत अच्छी ब्रांच है, उसके बारे में थोड़ा सा और स्टडी करो, थोड़ा एनिमल स्टडीज़ और हो जाए, तुम लोग बना के एक फॉर्मुलेट करके उसको दे सकते हो। ड्रग की लिस्ट बना लो, विच आर ओरल, which can't be which are IV, which can be not been oral due to some solubility issue or absorption issue or distribution issue, ये नहीं हो पाए। एक लिस्ट बना लो, उन लिस्ट में देखो कि कौन सबसे ज़्यादा मार्केट में इस वक्त चल रहा है, और उसके बाद एक लिस्ट बना के देखो कि अब उसका स्ट्रक्चर देख लो। स्ट्रक्चर देख के देखो कि तुम एक नैनो टेक्नोलॉजी के एक्सपर्ट हो, तुम एक फिजिस्ट हो, इंजीनियर हो, पॉलीमर के लोग होते हैं, जो भी एक्सपर्टीज़ पाई है वो देखो कि हम इसमें कैसे यूज़ कर सके नॉन टॉक्सिक लगा के। और कल को ये देख, एक बार एनिमल एक स्टडी कर लो, उसमें एब्जॉर्प्शन दोनों कंपेरेटिव कर लो, आईवी और ओरल को, विदाउट नैनो फॉर्मुलेशन एंड विद फॉर्मुलेशन ओरली देख लो, एक कंपेरेटिव देखो। आई कैन टेल यू, यू विल गेट दैट पेपर इन ए वेरी हाई impact journal if you love publication. If you don't love publication, keep with your heart, keep on working, do in the larger animal, and then send to the people and say we have a project, write a project, get some money from the people and say we have an idea, I don't want to show you compound, this is the compound, there are a lot of takers sitting. India is rich in making all those generic formulation and only formulating, they are only doing formulation work. So they love Cipla and many companies, they look for these idea. You float that idea, send to those people to write a letter and say I have a idea with me, I have a molecule, preliminary data in a mice shows something, I can't tell you which is the nanotechnology which we have developed which will be helping your compound, but this compound if I make it, if you give me 5 lakhs or 10 lakh rupees, I can do some animal studies and do a POC with you or a proof of concept, and if you really like I can transfer the technology and then we can deal further. For what initial work as an academic student or a academic teacher, I don't have money, you sponsor me, people are ready on the gate, they will, for them 10 lakh, 20 lakh, 50 lakh is not a big thing. If they get idea, they will make a lot of money and you will get a royalty of it. So these are the idea you have to think of it, okay. Access the niche and unique involvement. That is what I every time tell, don't go with a hi-fi thing, don't go on a fancy science. People go with a fancy sign, lot of English, I have a cancer drug, nobody has a cancer drug, okay, can't have in an academy a cancer drug until you face, you have seen now. You can only have an activity of a micromolar and you don't know whether it will survive to once it will given to the body or not. We have read all, now you have understanding how it goes. But you go with a small thing, that is what I am trying to say, access to niche and unique. मैं एक एग्जाम्पल दूंगा आगे चल के कि नीश और कोई बहुत अच्छा मॉडल हो, कुछ नया मॉडल तुम्हारे पास हो जो किसी के पास नहीं है, और इसके लिए पढ़ना पड़ेगा। आप जर्नल में देखो कि ये है, अच्छा इसका ये मॉडल है, कहीं हम ये मॉडल को हम डेवलप कर, ट्यूमर मॉडल है, इसको हम डेवलप कर सकते हैं, क्योंकि जीनोग्राफ्ट मॉडल है, हम डेवलप कर सकते हैं कि नहीं। पहले आपके पास एक पेपर वर्क हो, उसको पब्लिक है, उसको बायरक को लिखो, किसी को प्रोजेक्ट देने के लिए लोग तैयार है, बुलाते हैं सिबरी उबरी, वहाँ आके आप प्रेजेंट करो और नए आइडिया लो, पैसे लो। So that is a new idea. I am giving you all those some leaves for you guys to think of it and focus on it. That's all. You have to focus on small things and people are there to pay you. वो ढर्रा नहीं होना चाहिए, जो हमारे बॉस ने किया, बॉस ने बॉस को बताया, उसने अपनी ज़िंदगी में वही काम किया तो उसने उसको बताया, वो चलता चला आ रहा है, वही काम होता जा रहा है। अच्छी चीज़ें उनसे वो बेसिक है, मगर उसका एप्लीकेशन अगर हमने यूज़ कर लिया तो हम कहीं ना कहीं अच्छे पैसे भी ले लेंगे, प्रोजेक्ट भी मिल जाएगा, पेपर भी हो जाएगा और आपका सर्वाइवल भी अच्छा हो जाएगा क्योंकि आपको एब्जॉर्ब भी लोग करेंगे उसी तरह। ठीक। Establish a meeting, a platform and scientific forum, that what we are doing here. Regulatory infrastructure, the most important thing start a habit of confidentiality. In your idea, when you are doing some experiment और जहाँ भी बैठे हो एक डायरी अपनी पर्सनल होनी चाहिए, साइंटिफिक पर्सनल, ओके, उसको आप अपने जो भी दिमाग में आए उसको लिखने की आदत डालो। कुछ भी आए, कुछ भी चलते चलते आया और कहा यार अगर हम ये कर लें तो ये हो जाए, राइट इट, मे बी a rubbish idea, write it. And then if you get a free time, this is the time of a Google, this is the time of AI, this is the time you Google aloud, tell how I can proceed with this, and chat GPT, if you have something which is a detailed one, it will tell you how to do, at least it will give you a framework to work on it. And then you start, go to the library, work, start on it, okay? So important is the best way, that what I usually repeat a lot, best way to have a good idea is to have a lot of ideas. That is how we have to do it. This is academia versus industrial comparison. What in academic want is novelty, hypothesis testing, that is what their focus is. And industry is de-risking, हमेशा money उसके involved रहता है, इसलिए de-risking हमेशा डालते हैं. IND पेशेंट बेनिफिट, ROI कितना मिलेगा, कोई भी पूछता है ना, वो तुमने मारुति का ऐड देखा होगा, वो कहता है ये है एवरेज कितना देता है और कितना मिला, तो ये हम लोग का अक्सर फोकस रहता है. टाइमलाइन फ्लेक्सिबल, लॉन्ग टर्म, आपके पास कोई प्रेशर नहीं है टाइमलाइन का, आप 2 साल में करो, 4 साल में करो, पंचवर्षीय योजना बना के करो, कोई प्रॉब्लम नहीं है, बट आपके पास वो प्रेशर नहीं है, इसलिए आप उसको आयरन अप करके कर सकते हो, हैं, ये नहीं कि भूल गए और याद आया तो फिर किया, ठीक है, होती है, वहाँ लार्ज बजट रहता है और ये एक्सप्लोरेटरी काम होता है, हमेशा एकेडमिया का एक्सप्लोरेटरी चले तो ठीक, हमारे साथ ऐसा नहीं है, फेल फास्ट किल फर्स्ट, 80% ऑफ फेलियर अनरिलेटेड टू बायोलॉजी, दो बायोलॉजी में जो रिलेटेड अनरिलेटेड है, हमेशा फेलियर जो होते हैं वो पीके से आता है, टॉक से आता है, सॉलिबिलिटी डेवलपिलिटी. अब सॉलिबिलिटी कम है, कंपाउंड बहुत अच्छा है, मगर आदमी ये देखता है कंपाउंड की एक्टिविटी बहुत अच्छी है, एनिमल में भी बहुत अच्छी आ गई है, ह्यूमन में भी अच्छा है, मगर वो कमर्शियल एस्पेक्ट पे देखता है कि अगर हमने फॉर्मुलेशन में इन्वेस्ट इतना रुपया लगेगा, व्हाई द हेल आई शुड डू इट, दैट डजंट कम्स विद द कंपनी पॉलिसी और स्ट्रैटेजी, इसलिए वो रोक देते हैं. एनीवे, why focus innovation matters? Now the question is innovation. देखो दुनिया इनोवेशन पे जा रही है, दो काम होते हैं, एक होता है कि आदमी सब करने वाला जो है या परचून वो बेचता है, सामान बेचता रहता है और आप भी खरीदते हो, वो उतना ही कमाता है, 20 रुपए का लाया 22 रुपए का बेच दिया, यही उसका सिस्टम है, मिनिमम प्रॉफिट, मगर उसका रनिंग चलती रहती है. मगर किसी आप कोई डिज़ाइनर के पास जाओ, कोई होम डिकॉर के पास जाओ, आए और वो मुंह मांगे पैसे मांग लेता है, क्योंकि वो आपको पता है वो इनोवेशन है, नया है, नया के पैसे हमेशा. तो हमेशा फोकस करो नया, कुछ नया सोचो, है. प्रॉब्लम सॉल्विंग इज द बेस्ट थिंग विच यू कैन थिंक ऑफ इट, एंड एज अ साइंटिस्ट दैट शुड योर एम. डोंट ब्रिंग प्रॉब्लम विद अ प्रॉब्लम सॉल्विंग, or at least a leaf of it, discuss with a forum whom you trust or whom you feel that he will not humiliate you, and sit together and discuss. Usually आदमी सोचता है इससे बात करेंगे कहीं, बेवकूफ़ी की बात कर, चले यहाँ से. That should not be there, okay? Trust it, make a strong team, discuss openly that what we are going to do it, and hopefully it will help you out. Aligning with the industry need, sustaining academic relevance, ये सब. कल मैं लिंक्डइन पोस्ट देख रहा था, एंड दैट इज हाउ द चाइना हैज फ्लिप्ड द ट्रेडिशनल मॉडल, एंड नाउ चाइना इज सेइंग PhD स्टूडेंट नाउ नीड ए प्रोडक्ट नॉट ए थीसिस. ओके, तो मुझे पसंद आया, yesterday night only, today morning at 5:30 I put that thing. So what I am telling you कि अब जो है थीसिस नहीं, प्रोडक्ट की तरफ, अब एटलीस्ट चाइना से हम compete अभी आगे कितने साल में करेंगे, मगर हमारा थॉट प्रोसेस हो जाना चाहिए कि अब हमें कुछ ऐसा करना है जो कि आगे हम पेटेंट कर लें, कम से कम नहीं पेटेंट कर सकते, कुछ नया करके देंगे ताकि लोग उसको लीफ लेके पढ़ेंगे, ठीक है. और ये भी एक डिज़ास्टर है इंडियन इंडस्ट्री के लिए और अच्छी चीज़ है इनोवेशन के लिए कि फार्मेसी वर्ड जो है वो ज़्यादा दिन तक कॉपी नहीं कर सकता है, अब उसको नया चाहिए। अभी आपने देखा होगा इंडिया में यूएस में जो है, यूएस ने जब टैरिफ बढ़ा दिया और कितना इश्यूज हो गए, अब इंडिया के लिए एक मजबूरी हो गई है कि वो जो है अपना इनोवेटिव प्रोडक्ट बनाए। इफ दे विल नॉट मेक इट, then we all will be in pressure forever and ever. Okay, so now I think there will be a tangential change in the mindset of many people, even the governing funding agency, even the student professors and other people. There should be a change, and probably it will give a change. When you shake someone, then only things comes in order. Okay, okay, तो इंटरफेस स्नैपशॉट क्या है, what academia can offer, expert specific, identify and develop, state of art दे सकता है, identify and develop a technology, well trained human resource, patenting, licensing, tech technology, tech consultative, these are the adding point, targeted audience, stake. So what we can do is better, small funders and investor, venture capitalists की तरफ देख सकते हैं, साइंटिफिक रिसर्च सेंटर, दीज आर द पीपल होम वी कैन फ्रॉम academia point of view you can every time work with them and say these are the people looking for you if you bring all these things in the table. How in academia industry interface happen, upside of a अगर दोनों का कोलैबोरेशन हो जाए यूनिवर्सिटी और एकेडमिया का, तो उस पर फायदे क्या क्या है, increasing innovation and creativity trade, link scientific research results with the development, commercializing technologies, low unemployment rate, increase SMEs, SMEs you know small business and medium level of an employment engagements, and students have a opportunity to do that, a inflow of a talent, name and fame every time it comes. Once you have a name, what university need to work, capability slide. Now I think a university as a AMU, they should have one capability, whole capability slide, whatever they have they can put it, centralized list of a high-end instrument, best practice application. These if we do, then probably people will come, you have a half shot ready and you can do. I will tell you my personal collaborations and the story which I really love AMU, why I think AMU can also do it if other people can't do it. I have a collaboration with Harvard, with the Denisa D. Wagner Lab, and we have a Vistar University with Julia, Tel Aviv University by Danora. We have these collaboration going on, and what happened, they have a niche ACA model expertise which in industry need to prove of a concept, in return university or a PI get a good funding, say by the novel. This is how it is. What we do with them, they bring some niche. If we wanted some models, we wanted some animal studies, but they were having a ready. If we would have done with those guys, we would have done in a year, we would have established those method and models and all. So we paid a huge amount of money to them to help their collaboration, and then we allowed, given a tool compound to them to have a publication. Okay, because university says, oh, okay, fine, money is fine, but we need publication. From where we will get a publication? So that also get it. We have published some paper. This was the paper we published. See how many people are there in that publication. Okay, and this is scientific reports का पेपर है. हमने हार्वर्ड के साथ ये कोलैबोरेशन जब किया तो उनको एक टूल कंपाउंड दिया और एक जो एक्चुअल कंपाउंड था उसको हमने उनको हाइड करके दिया था ताकि उनको पता ना चले. And we asked them, you can publish with this number which is there, and they publish it, and this is the paper we got it. Okay, we also get the name in it because this is our molecule, and they get the paper. They published in they even सेलेक्टेड. एक और पेपर था जो हमारा, जो हमने जो है जो विस्टार इंस्टीट्यूट के साथ किया था, और ये भी देख लो कितने लोग हैं. तो पेपर ये नहीं है कि 2 आदमी का नाम आ जाए, 4 आदमी का नाम आ जाए, वो नहीं इंपॉर्टेंट है. कितने कंट्रीब्यूटर हैं, कितने रियल कंट्रीब्यूटर हैं, वो इंपॉर्टेंट. पेपर बड़ा हो जाए, नाम आ जाए, कोई वैल्यू नहीं है, कोई टेंशन नहीं है. अच्छा पेपर होना चाहिए, ठीक है ना. तो मैं फिर रिपीट करता हूं वर्ड कि साइलॉस पे रिसर्च नहीं होती है, इट्स ए कोलैबोरेशन. ट्राई टू रिमूव दैट बैरियर. We have to work together, and this, I was just trying to give you an example. I will tell you one story. This is also one paper we published here, and this was one of our drug which is still in phase 2 molecule, and this is our chemistry effort which we did it. And how means I am telling you what is the whole story, one giving one classical story. केमिस्ट्री की जाती है, केमिस्ट्री के बाद हम उसके SAR रन करते हैं, उसके बाद अलग सा उसका सेकंड आर्म को करते हैं, बाद में बायोलॉजी ऐसे करते हैं जो सेट होता है, उसके बाद बायोकेमिकल ऐसे करते हैं. वही कल उस दिन बता रहा था कि देखो ये IC5050 किस तरह से हम लोग करते हैं, ये पूरा जो है रन किया जाता है, फिर TR फ्रेट ऐसे है, दीज असेज इफ़ वी हैव. तो उसके बाद हम मॉडल रन करते हैं, करने के बाद जो है ये PD है, PD बायोमार्कर है, वो भी स्टडी की जाती है, और उसके बाद टारगेट एंगेजमेंट स्टडी की जाती है, जीनोग्राफ मॉडल रन किया जाता है, और उसके जीनोग्राफ मॉडल रन करने के बाद हम जो है फिर PK करते हैं, जो हम बता रहे थे ये रियल टाइम डाटा है और PK का, और उसके बाद PK प्रोफाइल में हम बताते हैं कंपाउंड कैसे काम कर रहा है. उसके बाद प्रोफाइल हो जाता है, ये कंपाउंड का टोटल प्रोफाइल है, लुक दिखता है. And then we do efficacy, in biology efficacy we do. This is the whole story which comes, and this is how the whole molecule package goes, and it goes to the IND, where we go and go to the FDA and ask FDA if they permit us to do that, and that's how FDA approved our drug to move forward. We did in US, we are doing in Australia, we are doing in India also. So this is the molecule, and luckily we have, I am too a member of that compound which has a patent on it. So what is the more important is a lot of work goes on a success of anything, and it takes a lot of patience, courage and effort to get it. कल मैं एक और चीज़ देख रहा था लिंक्डइन पे, और ये बहुत एक अच्छी न्यूज़ है. इंडियन पैंडोरम एक कंपनी है बेंगलुरु बेस्ड, जिसको कल उसने $10 मिलियन जो है रेस किया है फेज बी में, सीरीज बी फोकस पे. रीजेनरेटिव मेडिसिन के बारे में है, बायो प्रिंटेड ह्यूमन टिश्यूज, जो मैं बार बार बोलता हूँ कि फ्यूचर इज नाउ हेयर, फ्यूचर इज गोइंग टू बी ऑर्गन इन चिप, future is in going to do some in vitro where you can raise, or you don't want to use animal. People are pushing, there is ethic committee pressure on us that you should not use an animal. So this is the job of a scientist, to come up with the new ideas and say we have a model which is as good as an animal, at least 70% closer to the animal, where you are going to use 20,000 or 200 animals there, you can reduce to 20 animals. You have done extremely good job, and people are going to buy. These guys are also doing on those line funds for the operation subsidiary growth, and this is, I was trying to get this, I pulled yesterday only, and this is the news which came yesterday in my post. Okay, so these are the thing, people, there are two young birds and they raise a lot of money, and they have an idea, they floated it, and now they are doing it. So completing this whole process with the Stillness logo that let's unite to conquer the challenge for the patient to need. So this is how academia and the industry can collaborate together, they can make hand, and they can succeed. On this note, best of luck to you all, and hope you guys enjoyed it. Sir, stage is your all. If any question, please shoot. Of course, all those, see, toxicity is the only thing which doesn't need a specific thing to understand. You know, बॉडी का रिस्पॉन्स देखना है. अब आप सीधे पैरेंट मॉलिक्यूल दे दो, उसको किसी से लिंक करके दे दो, बॉडी के अंदर कुछ भी जाएगा, उसके टेस्ट में टॉक्सिक जो पैरामीटर वो सेम है. अब डिपेंड करता है कि आप किस जगह देखना चाह रहे हैं. टारगेट आपका लीवर है तो आपको लीवर और माइन्यूटली देखना पड़ेगा, आपको LFT करना पड़ेगा, उसको हिस्टोपैथ करना पड़ेगा, आपको देखना कहेंगे फाइब्रोसिस तो नहीं है, कॉलेंजियोफाइब्रोसिस तो नहीं है, हैं। तो हिस्टोकेमिस्ट्री में आपको उसको पूरे एंगल देखने पड़ेंगे। यहाँ आपकी लैब है माइक्रो उसकी क्या नाम पैथोलॉजी की, उनसे भी डील कर लो, उनसे कहो कि मैं एनिमल को दूंगा 28 दिन तक डोजिंग। वो भी डिपेंड करता है आप कितने दिन की डोजिंग के एनिमल डोजिंग आप सोच रहे हो। एक्यूट टॉक्स होता है, क्रोनिक टॉक्स होता है। एक्यूट में पहले दिन आप बहुत ज़्यादा डोज दे देते हो 1000 mg तक या किसी लेवल तक, और जाके देखते हो कि क्या हाल हुआ एक दिन में। फिर वो धीमे धीमे आप क्रोनिक पे जाते हो, 28 दिन का है किसी का, डिपेंड करता है कि साइकिल क्या रहेगी ह्यूमन में। तो आपने 14 दिन का पहले स्टडी करते हैं, हम लोग 7 डेज़ की रिपीट पहले करते हैं एक हायर डोज पे, और उसको देखते हैं क्या हुआ। फिर उसके आप टिश्यू निकाल लो, आप उसका जो है काइनेटिक्स तो आप नहीं कर पाओगे, अगर तो टिश्यू भी निकाल लो, उसको ऑब्जर्व करो, टिश्यू निकाल के दे दो किसी को, कह दो कि हमने लीवर टारगेट किया था और ड्रग हमारा, आपको ये कंफर्म करना है कि आपको ड्रग वो पहुंचा है लीवर में कि नहीं पहुंचा है। ये कंफर्मेशन के लिए आपको कुछ फ़ायदा हो रहा है कि नहीं, कैसे, उसके लिए मैंने बार बार बोला यू हैव टू डू ए कंपेरेटिव स्टडी विद एनिमल स्टडी एंड डू इट। HPLC अगर है तो अच्छा मेथड है तो मॉलिक्यूल डेवलप कर लो, एक मेथड डेवलप करके आपको पता चल जाएगा मॉलिक्यूल वहाँ पहुँचा कि नहीं पहुँचा, और नहीं तो उसके अंदर मॉलिक्यूल लेके आप किसी पैथोलॉजी वाले से बात कर लो कि ये मेरा है थोड़ा सा, इसका ज़रा सा लीवर देख के बता दो हिस्टोपैथ, कोई इशू तो नहीं है, हैं? तो वो आपको फीडबैक दे देगा, एक प्रिलिमिनरी इन्फॉर्मेशन हो जाएगी। फिर आपके पास पैसा आए तो देयर इज अ लॉट ऑफ टॉक्सिकोलॉजिकल लैब अवेलेबल, दे विल टेक सम स्मॉल अमाउंट ऑफ मनी एंड प्रोबेबली वंस यू कम ऑन एकेडमिया दे मे नॉट टेक यू मच ऑफ ए प्रॉफिट एंड दे कैन डू योर स्टडीज एंड दे कैन बी हेल्पफुल फॉर यू। Anything else? Done? Good. Yep.
And if is there any
I
ethical...
missed you. Can you come again?
Sir, can you tell about procedure that how a novel idea reached industry directly? And if is there any ethical guideline approved by FDA regarding this?
There are two things. I should repeat, rephrase your question if I understood rightly. You want to say that how in industry you come up with novel idea,
Yes, sir.
or you want to say that you have a novel idea, how it will come to industry. Which part you want to know?
Sir, my novel idea
Wonderful.
that should reach industry directly.
Absolutely, it's not directly. That's what I was telling. I told you earlier that even given a nanotechnology example, then you have to first do some preliminary work. You have an idea, everybody has an idea, but this idea, you conceived the idea, but idea is really going to work or not. So at least some preliminary hypothesis you should have. If you have a preliminary hypothesis, you have worked something, you have some data with you, and then you can write to the companies which are working. If you work on, there are a lot of patent are going to expire, okay, this year or next year. See those molecules. A lot of Indian companies started working on it. So you think of that and limit yourself and say, इसमें ये issue है और हमने इसको सॉल्व कर लिया। मान लो एक हम एग्जाम्पल, बेस्ट एग्जाम्पल तुम्हारे ओरल बायोबिलिटी का दे देते हैं, इसकी ओरल बायोबिल नहीं है, तुमने कहा मेरे पास आइडिया है, अब मेरे पास ये मॉलिक्यूल, मेरे पास जो हम काम कर रहे हैं, अगर हम इसको अटैच कर देंगे, क्योंकि इसमें क्लोरीन वॉट एवर द अटैचमेंट नीडेड उस स्ट्रक्चर में है, वो काम कर जाएगा। मगर मेरे पास पैसा नहीं करने के लिए, अगर आप मुझे 1 लाख 2 लाख रुपए दे दो अर्ली ऑन सीडिंग फंड, तो हम आपको ये प्रूफ करके बता देंगे, और फिर बाद में हम और पैसे लेंगे और काम करेंगे। ठीक है, आप उनसे एक CDA साइन कर लो, कॉन्फिडेंशियलिटी डिस्क्लोजर एग्रीमेंट कहते हैं, उनसे साइन कर लो, तो दोनों के बीच में कॉन्फिडेंशियलिटी रहेगी, वो आपकी बात को फैलाएंगे नहीं, बात करेंगे नहीं। और दूसरा ऑप्शन वो कहेंगे हम आपको एब्जॉर्ब कर लेते हैं अपने यहाँ, ये भी एक ऑप्शन दे सकते हैं, और को नहीं मैं खुद करूँगा, अपनी PhD करूँगा, आप ये फ्लोट करूँगा, तो इट्स एन आइडिया। तो डिपेंड की व्हाट यू आर डूइंग, यू हैव टू राइट, यू हैव टू रीच टू द राइट पीपल हु आर रियली इंटरेस्टेड टू दैट। Okay, and the other way is the forums. Go and join some conferences, present your paper and say that this is what I have done, I can't open many things. I have a confidentiality with it. Don't open every damn thing which you have. Just closer yourself, put some card on your chest and show something which can attract people and say, I can discuss you. Let's have a CDA with you. Okay? Yes. Anything else? Good. Thank you very much. Appreciate. Okay, I will be leaving.
Okay, so thank you so much.
Hey, thank you.
And if you have any questions for him, he's available for a few more minutes, I can say.
Yeah, yeah.
And if not, then again, we are always connected. I'll share you his email and phone number and everything.
जो हमने कुछ अगर फंड लिया है, अगर सर हमारा डिज़ायर रिजल्ट नहीं आता है तो उस फंड का मतलब कुछ हमें रीपे करना होता है, या जैसे हमारे पास कोई आइडिया है उसको हमें इम्प्लीमेंट करना है, उसके लिए हमने कोई सीडिंग राउंड रेज किया तो उसके लिए हमने कुछ फंड लिया पर हमारा डिज़ायर रिजल्ट नहीं आया तो उसके बाद
तुमने कोई गारंटी दी थी उनको? हैं? Leave it. This is all seeding. It's all. They have not given you $1 billion for that, na? This is a seeding. It will work. It will pass or it will fail. It will succeed. You don't know. We are all doing experiments. Everybody understand that, huh?
All right, so we can take a 5-10 minute break before we go on to the last session, lecture 14 with me. And then you will have some time to review and prepare and give us the feedback, fill up that feedback form. Then we go for lunch, and soon after lunch we'll have the exam. Does that make sense? You'll have some time to prepare and got all the material with you, right? And to fill the feedback form. Just take 5-7 minutes vitamin D or tea or whatever you want. Then we'll be back. Okay?
यही रख रहा हूँ, ठीक है, कार दूँ यही रख दो। जो एग्जाम होंगे वो दिखा सकते हैं, एग्जाम होंगे वो दिखा सकते
नहीं, आपको पता होगा, दिखाते हैं क्या?
मैं पिछली बार भी हुआ था इनका, कुछ भी दिखाते हैं इसमें, उसमें बस लिख देते हैं।
ठीक है, आप उस टाइम अपना अपलोड कर लीजिए, टाइम लिख दीजिए कि एग्जाम गोइंग ऑन,
वैलिडिटी लास्टली, और सर एक और चीज़ सर्टिफिकेट देगी एग्जाम का, तो वो ऑन द कैमरा दे देगी, ऑफ द कैमरा दे देगी,
ऑन द कैमरा देगी,
ऑन द कैमरा दे देगी, उसमें कोई ब्लैंक दिखाना, वही चीज़ है जो भाई ऑफ द वैसे ही पकड़ा देते हैं बाद में।
नहीं नेसेसरी है कि सिंपल दे दीजिए।
हार्ड डिस्क तो आता है उनका, मेरे पास तो पटेल
Is that better? Okay. All right, so we are moving towards the last session, last lecture. And there were a total of 14 lectures, plus the tutorial that you had. So this will be the last one, but to be honest, I learned a lot from this course. I got to know about your enthusiasm. I got to know about a lot of things that I didn't know before. So I hope you also had similar experience, and you will be able to translate into something tangible. I always say that try to come up with something that tells you that, yes, I have done this, right? So productivity is important. All the industry people are just saying productivity, productivity. We have to see productivity within ourselves as well. So I'll be going through some of the lessons that I learned, and I think it will be helpful for you as well. Although there was too many things that were squeezed or compressed into these 5 days, right? There were multiple things, some of the things that you would have seen or heard for the first time, some of the things that you might have already heard about. So it's difficult to synthesize everything, like understanding all the clinical trials, or understanding formulations, or understanding target identification, or understanding the mechanism. But it does give you one basic thing that I would say, whether you are interested in taking up drug discovery as a career or not. That's the first filter. Some of you would identify that, yes, I can, and this is my field, this is my passion, this is where I belong to, and this is where I would go. Right? That's the first filter. Second, if you think that, yes, I should be into this drug discovery and development and delivery industry or field, then you can identify yourself, where do you belong to? Because now this is all about expertise. As you are advancing into your careers, it's all about expertise. You can't be jack of all trades. You say, I will be manufacturing my stuff to getting the Nobel Prize, and everything that I will be publishing will be one day a Nobel Prize stuff. No. In science, in PhD thesis, there's a very small aspect of a particular problem that you are able to solve, and that becomes the thesis. So you go towards specialty and super specialty. So I hope you had some idea, a broader overview of academia, industry, company, patents, formulation, nanotechnology, and target identification, understanding the disease mechanism. There's so many fields. So you can subclassify yourself that, okay, this is the one that I want to do. That's I'm passionate about. So a lot of stuff was thrown onto you. Now it's up to you to see where I belong to. And that's how I'm going to take into my next career, right? If this is only if you think that, yes, I want to be in drug discovery and delivery field. So can I just ask you, what was the most important lesson that you got? You don't have to answer now. At the end, I would be asking this from all of you. Right? So all of you would just try to synthesize it in some words, that this was the most important lesson, just one. So, and I'll be giving you some hints here. You might repeat that as well. Number one, from my first lecture, I'm emphasizing on this, and that's why the topic is also drug discovery and delivery requires multiple expertise. Even on the first day, you guys saw, there are so many people who come together to make a successful molecule or a drug or able to cure a disease. It's nobody, I mean, no matter how good you are, you might be a very good molecular biologist. You might be a very good formulation scientist. You might be a very good chemist. You might be a very good IP person. You know how to patent everything right, left, and center. So you can be good at something, but you cannot work alone and develop a drug. It has to be complementary. It has to be multidisciplinary. Several fields have to come together. So that's the lesson number one. And mostly it comes from academia. Academia means universities and colleges and institutions where teaching is going on, hospitals where the actual problem or the actual customers are for the drugs. Drugs are given by whom? Physicians. Physicians belong to hospitals. So that's why you saw in one of my slides, there was bench to bedside. So far, it's only from bench to bedside. There's no feedback mechanism. Hardly physicians have any time even to talk about their experiences with the patients. But now, the concept of physician scientists, that's picking up. So I know several MBBS from here only, they have now their own companies. Somebody is working on a small gadget, somebody is working on a AI-based company, somebody is working on a drug discovery company. They are not seeing patients, rather they are using their patient experience to develop something that can be useful for them, right? And if you can't do it by yourself, if you're not a doctor, you can at least talk to a doctor. And I also give you an example of my consortium that we have got clinicians, we have got molecular biologists, people from engineering. We all meet every month and we discuss problems that, okay, what is your experience about the patient? What does patient actually need? And sometimes the ideas come from their experience that we saw this, and Sayed Saab, like the chief guest, he also told you that if we can develop a device which can deliver the drugs intradermally, it will reduce the dose by 1/10, right? So this is something that is coming out of the experience, not from the textbook, that people are more compliant and they would be taking this drug in lower doses. So that's the lesson number one. And yesterday, the exercise that we did was, one of the reasons was to make you talk among yourself. Otherwise, usually you talk with your friends. You talk to your department people. You talk to somebody whom you know. So I remember when we used to go in conferences in US, so one time they tagged our coats with colors. And then they said, we want to see a table during the lunch filled with as many colors as possible. So you're coming from zoology, you're having a different color. You're coming from biology, you're having different color, engineering people. Let me see a table which has got multiple colors, diversity. Right? So those are the things that are needed to encourage this kind of, and here I differ from industry people. Because industry people, they say confidentiality. When you're doing confidentiality, you are keeping things to yourself. How can you spread out? How can you talk about these things? So now you have to be smart enough to conceal the core, but at least open enough to collaborate, right? You don't have to tell the exact molecular structure of what you are thinking about, but yes, you can talk about its effect. You can talk about the problem that it can solve. You can talk about its target, right? So I'm not against, rather I'm vehemently against not sharing. The whole world is talking about sharing data, whatever you are generating in the lab, talk, come together. And industry people, no, hide. Right? I even challenge that if my idea, if somebody can take it and do it, I'll be very proud of that. So that's a perspective issue. So collaborative ecosystem is very much needed. We are trying to build it here in AMU. I know there's a lot of resistance. It needs a disruptive mindset, but inshallah, it will be there. And it also has to encourage open innovation platforms. Like, suppose I have to talk to you about your data. How would you share? You'll either bring your notebooks, right? But it can be only just two people talking here. So electronic lab notebooks, they allow you, there's a shared repository of the data. In the Cleveland Clinic where I used to work, there was a tool. If I needed some reagent, I just post it in the group, and everybody knows, okay, this person needs this reagent. Whoever has it, they can borrow it, or they can give it to them. Or if somebody, like the platforms like ResearchGate, right? You post your questions there, and there's a lot of people who can see your question and answer it. It's very helpful. Has anybody tried ResearchGate? Okay, so ResearchGate, I think it's a very nice platform for doing it. There are many other platforms, but within our university, we don't have any such thing, right? So we can have some kind of, you can start it in your department, you can start some discussion clubs, journal clubs, where you can come together and share some data on a periodic basis. We do it, and we have a coffee club where we just talk about science 1 hour in a week, share some data, see what you're doing, where you're stuck, how I can help. So you're not having a journey alone. All right? So that's lesson number 1. Can you say that with me? What's the lesson number 1? Collaborate. Talk to each other. Have that multidisciplinary mindset. Don't feel insecure. What do you see here? Anybody can guess what is the picture about? Yes, it's a list of authors. Have you seen the human genome paper? That was my first instance where I was new to the field, and I was trying to understand whether this is a paper which is having some substance or only the author list. It was several pages. And this guy, he's a big name in science, Eric Topol. He got the Ig Nobel Prize. Nobel Prize we all of us know about, but there's something called Ig Nobel, IG Nobel. It's just opposite. Why? Because he had how many authors there? In 1993, he had 972 co-authors. A very good journal, New England Journal of Medicine. It's a highest rated journal in medicine. Impact factor 37 and above. Probably more now, it could be 50 as well. But he had so many co-authors. And the latest record is by some physics people. They have got more than 5,000 authors. So they are the top numbers where one paper is being authored by more than 5,000 people. So don't hesitate in giving away the authorship if somebody is contributing. No decorative authorship. So, right? So don't feel that insecure that why I should be giving more. Rather, it tells about your collaborative potential. It tells that yes, you can collaborate. So gone are the days then when you write a single author paper and say I am the authority. It's taken negatively. That means you don't have the ability to work with anyone. Rather, if you have got more people, but there has to be balance, otherwise you will get the Ig Nobel Prize. Right? Not the Nobel, rather the Ig Nobel Prize. So there has to be a balance, no decorative. There are full guidelines on who should be getting an authorship, who should not be getting an authorship, so I'm not going into that. But yes, this is possible, people are doing it, so don't hesitate in giving away authorship. Like we just discussed about this group, that if suppose, I'm presuming that all the participants are coming on board to write this review, how many will be there? We have got 58 plus 3 of us as the faculty members, so 61 authors will be there on this review. I know it's not going to happen, but still it will be a huge number. Right. Lesson number 2. We have done this exercise yesterday that target identification and validation is a separate field. And I think that this is where the whole story starts. Unless you know where and whom to target, so there's no drug discovery going forward. So first thing, and this is close to my heart because I do as well, so I'm a little bit biased, that lesson number 2, work on the mechanisms. That what are the molecules that are responsible for causing this disease, or even more basic. Now, the trend is that the world is moving towards longevity research. People are working on how to make this aging more affordable, better in terms of the quality of life, and a lot of people are getting funded for that. So those basic research stuff is important, and we need to get into finding those mechanisms, those molecules, those pathways, those interactions that are going to give us the target that will be moving forward in the drug discovery, right? And definitely, this is an entire career that people make out of it. Robust validation pipelines, that if you say something that P53 is involved in cancer, you have to prove it. And you don't have to prove it just by 1 or 2 or 5 experiments. Rather, you have to prove it in multiple ways, in multiple instances, and over the time it has to be tested. One thing here, there are some flukes where there are some discoveries that are just, once they come up in the news, and then they die down. I remember there was a news that came from Israel that there's a drug that they have found for cancer. Just one news item, nothing before that, nothing after that. It was a fake story. Another story was about nitrogen oxide synthase. Somebody reported in the journal Cell. Cell is a very good journal. They said that they found it in some plants, a variant of nitrogen oxide synthase. That one story, nothing before that, nothing after that, it was finally retracted. It was a fluke. So whatever you are doing, whatever you are identifying, the time will test itself and people are going to experiment on it. So have some robust testing methods. Be brutal to yourself. Don't just start jumping on one particular idea. I found it, I got it. Now test it in multiple ways, statistically as well as qualitatively. Then only you say that yes, this is the target. Right? And that's why I love this multi-omics approach, because it tests you from different angles. It tests your target from different angles. It tests it from different biological perspective, and that gives more confidence without any kind of bias. Right. This does not mean that, I think this is another contradiction that I have with the industry, because in academia, publish or perish is the model. You like it or not, if you need to get your PhD, you need to have papers. If you will be applying for postdocs, what will you be asked for? A product or patent or paper? Papers. China might be talking about it should be a product rather than thesis, but sometimes you enjoy doing discovery. Just this Tim Allison, who got the Nobel Prize for immunotherapy, he was working on immunology. He was working on antibodies. He was working on tumor cells. He was talking about cell-mediated immunity. That eventually became the immunotherapy for cancer. But in his Nobel Prize speech, he said, I never thought about it. I never thought about any application at all. I was just trying to understand the interaction of the T cells with the antigens, the antibodies. I was just working on the basic biology. And that's why Nobel Prize is given only for basic science discoveries. So this is as important as thinking about a product. So I'm not denying what my colleagues have said. They are having their own perspective, but don't get demoralized if you are passionate about finding out some new mechanism or studying a particular molecule, which is having no relevance, no application today. It might be tomorrow. There would be a day when people will realize that this is implicated in some kind of a disease or a condition. So publish as much as you can. Right? So lesson number 3, because I was given this mandate of also suggesting the framework in academia, that how can we develop a framework that would be helpful for the drug discovery process. So in that, we do need a lot of infrastructure and technology access. Thankfully, we are not bad in that in AMU or even in India. We are having good hubs where you can get the access to the technology. We are having clusters here where you can do a lot of computational stuff. We have got labs, we have got parks. They were talking about C Camp in Bangalore. They even have the incubators facility. If you have an idea and you say, I don't have a centrifuge in my lab, or I don't have this in my lab or that in my lab, you can go there and incubate your ideas. This is where the incubators come from, which means you don't have to start building a new lab or looking for a lab which has got everything. Rather, you go and incubate the idea. They will provide you with all the support. So you don't have to worry about buying reagents or buying the equipment, or even they help you with finding investors. right? So this is called the biotechnology parks. North Carolina was the first one, I guess, which started this concept. So they built the facility. All you have to do is to pay a fraction of the amount that you have raised in funding. Suppose you had an idea that this is who's Nicola Sativa? You're the one, okay. So you have this idea. Now you went for some funding request. You got, say, 50 lakhs, right? Now with this 50 lakhs, what would you do? You have the option of going and buying your reagents and equipments and then doing your own research, right? This is how classically it is done. Or you can pay an incubation facility, 5 lakhs. They provide you with all these things. You get things done fast, and you get things done in a professional way. And then if the results come in, you are ahead in the game. So this is what is needed more efficiently being done. We are also at AMU coming up with the incubation center where you'll be able to incubate your ideas, right? So this will have all the wet lab facilities as well as AI ML tools. Now again, no institution or no country can have all the facilities in one place, right? You might have to again do what? Collaborate, exactly. So you might not have everything in one place. You'll have to collaborate with the people who have it. Lesson number 4, funding and IP management. See, we are not wired for thinking about how to raise money. We came into science, we wanted to study, we have got a mindset of studying, analyzing and all that, but we don't know how to make a sales pitch. अगर सेल्स पिच होती तो फिर हम शायद कोई सेल्स वाला कैरियर पकड़ते, साइंस का कैरियर ही नहीं होता. We would have gone into management or something. So we are not wired for that. If we say we have an idea, give us 50 crores, and then we promise, and so you might have seen the Shark Tanks and all. But it's, to some extent, it's necessary. We'll have to learn it so that we can at least communicate our ideas properly. Don't overblow it. I've seen people who are promising moons and stars and then delivering nothing. This will not happen many times. If you fail honestly, it's fine. That's okay. But on purpose, you promised a lot of things. My molecule does this and that, and it will cure cancer, it will cure this, it will bring you 50 crores of revenues. If you're doing it intentionally, today or tomorrow, you'll be found. People will know that you are faking it. So be honest with that. My philosophy, which does not fit in today's scenario, is promise less, deliver more, right? But that's also not correct. At least promise as much as you can deliver, right? So this is important in terms of funding, getting some funding and secure your ideas through the patent or licensing, or even in some cases you can just talk to a lawyer that how should I protect my stuff. If patenting is very, very expensive, so they'll find you ways on how you can protect it, right? Then we also have to go into translational pathways and regulatory alignment. So we need to know that, okay, in my PhD thesis or in my master's project, I did this work. And with this work, what is the next step? How am I going to really translate it into a company or some kind of IP? So people have got different choices here. You might start with a product. You just want to develop it to a level where a bigger company comes and acquires you. A lot of people are doing it. And in US, this is like in Boston and in California as well, these East Coast and West Coast, they have got these hubs. I know several scientists in Boston who are spinning off companies. So they do something, they're working on a pathway, they found a target, they spin off a company. And now this company is taken up by Merck or bigger companies. They acquire you for a huge amount of money, and then you exit. Start working on a new problem. So again, you have to think about those strategies on how far you want to go with your product. And this is very difficult where you think about your molecule or you think about your drug or you think about your target and you take it to the finishing line. It doesn't happen in a lifetime sometimes. So if you identify a target, if you identified a molecule, if you found something, you might want to give it, it's like a relay race. It's not a marathon. Relay race means passing the baton to the next person. So you have to think about it if you have got the long-term vision of how am I going to translate it to the next level. So that's your responsibility, and then only you'll be able to find the right people to talk to. So there's a need for setting up the translational research offices. This is not existing for now. At least I don't know anywhere in India where people are having a center for coordinating the things from lab to the clinic. Like I have 2 PhD students, and one of them, she is now working with the clinicians to get the patient samples, and that's the only way we know here. And then those patient samples come to our lab and we do the molecular analysis. But if there is such a coordination center where the lab can connect to their clinic, that will be excellent, right? So that's not your responsibility. Some of these take-home lessons will be going to the policymakers as well, that they have to think about developing these things for better drug development and discovery. And definitely we have to follow the regulatory framework. Every country has got their own regulatory framework. You have to be compliant to that. Make sure that you're not stepping on somebody's patent. You're not stepping on doing something that is not allowed in that particular territory. So be careful about your research, that you're not violating any kind of rules or regulations or legal things that might land you in trouble. I know one of the biotechnology companies in US, he was an agricultural biotechnologist. They had a transgenic crop. So the transgenic crop was wonderful, idea was wonderful. He just planted it in a field for testing. And that had adverse effect on the environment. And then he was sued, and ultimately the company had to be shut down. So the regulatory compliance is very, very important. Again, not your job entirely, but you have to have some legal experts that can help you with that. Don't lose sight of it. Even here, it's okay. I've seen people draining their chemicals in the sink, right? I don't know how many of you have done it. You don't have to raise your hands, but this is the common practice here. But in US, if you're found, you can be jailed. Acetyl bromide down the drain. You can't do it. I remember once I broke one thermometer in my lab, and there was a whole team that rushed into the lab, wearing all kind of suits. I said, why? They said, this is mercury, don't you know? I said, we keep playing with this in my childhood. We used to play with it. He said, what? This is mercury. And the entire team with suits and everything, they were there to pick up that small mercury. Yes, it is hazardous, but the way we work without gloves, without apron, I'm sure once you reach to those countries in a good lab, you will find कि हम क्या कर रहे थे पूरी जिंदगी. कोई सेफ्टी नहीं थी, कुछ नहीं थी. ना गॉगल्स होते हैं, ना ग्लव्स होते हैं, ना लैब कोट्स होते हैं, ना शू कवर्स होते हैं, ना हेयर नेट्स होते हैं. हो रही रिसर्च. So don't lose sight for that regulatory alignment and translational pathway. Therefore, there are some guidelines like good lab practices. It's called GLP facility. They ensure that the environment in which you are working and developing your research is compliant. Right? ये नहीं कि जब आप protein पे काम कर रहे हैं, you're rubbing your skin and the keratin is coming off, and this is what you're seeing in your mass spec analysis. You say, wow, I found some good results. No, it's keratin, right? So don't go into that. Develop this habit, at least a mindset, that you would be doing the good laboratory practices. Then there's also good manufacturing practices. That's not your domain, but think about GLP practices. And this also involves data keeping. I think people have already talked about it, that you should be writing down everything that you do. again, we don't reach to that level. When I went for my postdoc in Cleveland, I was given an orientation that this is how you should be writing your notebooks. At that time, it was not electronic. This is where you sign, and get it signed by a supervisor, and get it signed by somebody else who has done it. If you don't have those things, you might land in some kind of legal trouble. And we think that, no, we are not going to upgrade science. What happens? So develop these good habits of doing science of international standards, and that's the whole purpose of this course. That don't look around yourself कि मेरा दोस्त ऐसे ही करता है, मैं lab coat पहन खड़ा हो जाऊँ, people will laugh, and this happened to me. When I came new, because I'm just new in Aligarh, just 4 years ago I came, and lab coat, goggles, and gloves. They're laughing. Namuna kahan se aa gaya? But it's not that kind of stuff. Maintain those standards. At least you should know how things are done outside. Right? Data keeping and writing notebooks. Notebooks maybe, if you're tired by the end of the day, I suggest to my students, at least record your voice. Keep a voice recorder, or if you have got a high capacity phone, you can use it for that as well. That record your entire day observations. I saw this, and whenever you get time to write it down, do that. Right? Lesson number 6, cancer drug development is complex and multidisciplinary. This is more of an advertisement slide because I work on cancer. So that's the lesson number 6, that don't underestimate the smartness of these cancer cells. They're really, really smart. And as we are getting to know more about them, we are knowing that we don't know anything. They have got, like AMR, antimicrobial resistance, they are even smarter than bacteria. They know how to live without food, they know how to live without oxygen, they know how to live without any kind of blood supply or nutrition. So we need to, if anybody is wanting to work in cancer drug development, they should be ready for this acknowledgement that it is pretty complex and it can be handled by one person. And I have put this slide for you to discuss. That the fundamental knowledge is biology. Even I'm teaching nanotechnology. I teach the fundamentals of nanotechnology. But I tell them that Richard Feynman, the guy who is supposed to be the father of nanotechnology, he also thought about protein as a nanoparticle. So biology is predominant science. It's debatable. We can discuss that. But think logically and think biologically. That will give you a lot of aspects about chemistry and physics. So how many of you agree with this slide? Can you raise your hands? Okay, I'm looking for someone who doesn't agree. So you don't agree? Okay, can you give me your idea about that, why you don't agree? In the middle. So you agree with this? You don't agree. Okay, so let's hear why. I think I can have that mic somewhere.
Sir, I think it was physics and physical forces which led to the development of life on Earth. So it was first physics that led to the development of biology on Earth.
Okay, that's a good perspective. That the origin was because of the physical forces. All right, anybody batting for chemistry? No? So physics I agree, and then physics and biology, okay. Chemistry, so we put a reason for that. Yeah, also is fine. Physics also, biology also, chemistry also, this is totally fine, I agree. But here it's written The basic science is not physics. It's denying that physics is basic science. It's denying that chemistry is basic science. It's saying that biology is the basic science. Just think about it, and you can discuss it yourself over lunch or coffee or something. And the most important lesson, I think this is not a lesson from the drug discovery, but this is a life lesson which would be helpful, and I thought of sharing it with you. So focus. My boss, he was an Irish American, and he always, and I was when I started my postdoc, and when you go from India to US, you're jumping with ideas. Ye bhi kar loon, wo bhi kar loon. This is great. I am good at this. So then he said, focus. Focus yourself on one particular thing and try to take it to the finish line where you can assess whether you want to go further or not, rather than being someone who's trying a new idea every time. Right? So it's not good to be diffused. Rather, your focus will definitely help in your careers. And the second thing is discipline. I'm sorry to say, but this is something that we desperately need. Discipline in terms of work ethics, discipline in how we are conducting our research, or discipline once we reach to a company where we'll be doing all this work. So this could be a make or break situation. So without discipline, I don't think industry people will even let you go more than a year. So discipline is very, very important. And there's no replacement to hard work. People might tell you that there's something called smart work. I don't think there's anything smart work without hard work. There could be smart work with hard work, that's totally fine. But hard work is the denominator. Without hard work, you think that in 2 hours I will come to the lab, run a gel or run one experiment and go, and tomorrow I'll be a great scientist. No. You have to burn the night lamp's oil. You have to be doing experiments, reading, analyzing, thinking, discussing. There are a lot of things that you need to do that all make up the hard work. Right? And I mean, there could be many here, but for me, I found this to be relevant for this class. Consistency. Because in our field, we see a lot of failures. We don't see success very often. And remaining consistent, motivated, and doing things with a conviction, a dedication, that's important. So they say rest if you must, but don't you quit. So you might feel demotivated, experiments fail, but if you're consistent, you will be there one day. This, I think you have seen the examples that he was here, Dr. Imad, very experienced scientist. So this, what brought him here is consistency, focus, hard work, dedication. So these are life lessons, but you can apply it wherever you want. I think I don't have any more slides, but yes, I have to say that this was a great class. The way you interacted and the way you came up and the way you have been good students. And I hope you can impart this to let it amplify. So each one of you can be a mentor as well. Don't think that once I become a faculty member, then only I'll be a mentor. No, you can mentor your school students, tell them about all these things. Not the exact science, don't start telling them the structure of benzoquinone, but at least you can talk to them about these basic stuff. All right, thank you for patient listening, and let's warm up with discussion. First thing is to, thankfully we have got internet. First thing is to go on their website and see, right? And then I know the procedure that they have got calls where they ask you to submit your proposals, and they tell you that we'll be giving you 5 lakhs, 10 lakhs to fund your proposal. Then they have got the machines that you can go and work with, and
Are they having infrastructure for that, where we can work there?
yes, they have got infrastructure. They have got machines as well.
We are not aware of that.
Yeah, so that's why I'm saying go to our website and see what is available there. If it is relevant for you, you can apply. And there's application procedure as well. Yes, anybody? All right, so if we are done, so I will be leaving you guys with the questionnaire forms and the time to revise. We will reconvene after the lunch at 2 for the exam. Right? Meanwhile, I'm around, so if you have any questions about the course or about the slides, I'll be happy to answer that. All right, thank you. Let me distribute the form. Let us do it. And your grade sheets will be provided to you electronically, but certificates will be given after the exam. So hang around, don't run away after giving your exams. You'll be given the printed certificates after the exam. I think you can just no, there's a grade sheet as well. So this is a 1 credit course that you'll be graded on. So you'll be getting grades A, B, C, D after the exams, right? That we'll be sending electronically because we can't do real-time paper checking and marking. That we'll do later on. All right, so we take the break, prepare, and we come back at 2. But we'll start sharp at 2, and there will be 50 questions for you to answer out of 60. They're all multiple choice. You do with a pen, write your name on each paper, and you can just mark. Try not to overwrite. If you're doing overwriting, just do it clearly so that we don't misunderstand your options. Clear? 50 out of 60 questions you have to do, and you just have to take it with the pen. There's no OMR filling sheet, no. Just put a right next to the correct option. There's no negative marking as well. And don't forget to put your name on every sheet. All right, so we are now towards the concluding ceremony. Can you hear me properly? We are towards the concluding valedictory ceremony, and for that I'm very honored and glad to have Professor Varsi with us. He's the GIAN coordinator, and his efforts have led us to this point that we are able to conduct this course and conduct it successfully. Professor Varsi, I would please like to request you to come over to the stage. I would kindly request Dr. Kasi Anwar to present the potted plant. So, looking into the sustainability mandate जो हमें मिला हुआ है, तो हमने बुके वगैरह के बजाय ये चीज़ शुरू करी. So this program, this entire course, the focus is on the participants. I'm not saying students, but participants, because some of you are accomplished scientists or doctors, or so you are already having made your career in that. So I would like to invite a few of you here and say a few words about how you found this course useful. आपको क्या चीज़ें इसमें सही लगी? So please, any one of you can come and speak, please. Can you introduce yourself, please?
Assalamu alaikum everyone, respected guest. And I am Dr. Samima Khatun from Department of Chemistry. I am recently doing as a guest faculty in the department. So actually I am congratulate sir and the whole INC department for successful this 5 days program. It was very wonderful to attend such program, because the reason is, so I am feeling personally that I am attending 2-3 conference in the same time, because I obtained the scientific academic knowledge from Imadul sir and industrial perspective from Jainuddin sir and Sayyid sir. So it was a great pleasure for all of us, and also the audience is from all departments, from biochemistry, from microbiology, botany, Unani medicines. So it was really a multidisciplinary approach, and because I completed my PhD in 2019, so after that I involved in teaching, but along with teaching I personally doing some research along with, but somehow it is somehow slow this time, because along with teaching it is difficult somehow to carry my research. At first it was my research time, so I published in my research time I published 2 papers each year, but this time in 2 years 1 paper, so it is somehow slow. But after seeing Imadul sir at this stage, the enthusiasm he is putting in research and drug discovery, I get a fresh restart or gear up my research after attending this program. So it is thank you sir for giving us this opportunity and for this program, so that we can restart our research afresh. Thank you so much and congratulations.
So anybody else would like to please come?
Assalamu alaikum. Professor M.J. Varsi sir, Professor Dr. M. Azhar Aziz sir, my dear fellow participants, guest sitting on first row of this assembly. So very good afternoon to one and all. So सर के रिक्वेस्ट पे, अजहर सर के फीडबैक देने के लिए मैं आप लोगों के सामने हूँ. पूरा इवेंट तो जाहिर है 5 दिन का है, उसका हर सेशन का फीडबैक देना तो थोड़ा मुश्किल है, पर ऑल ओवर जो प्रोग्राम था, पहले तो ये बताना चाहेंगे कि आई एम डॉक्टर फहमीदा जीनत, असिस्टेंट प्रोफेसर इन द डिपार्टमेंट ऑफ गायनी एंड ऑप्स, विच इज़ मेंशन एज अ निस्वा व कबालत, अजमल खान तिबिया कॉलेज, फैकल्टी ऑफ यूनानी मेडिसिन. So I'm the basically from the side of Unani medicine and especially from the clinical field, gynae and obs. इस कोर्स को ज्वाइन करने का हालांकि हमारे फील्ड से हटके है, लेकिन कहीं ना कहीं से अटैच है. ज्वाइन करने का मकसद था कोलैबोरेशन, अंडरस्टैंडिंग ऑन द बेसिस ऑफ नैनो टेक्नोलॉजी. हम अपनी फील्ड में अपनी दवाओं के साथ अपने स्टूडेंट्स या हम खुद इनके साथ जुड़ के किस तरह से अपनी मेडिसिन्स को नैनो टेक्नोलॉजी के फील्ड में किस तरह से और डेवलपमेंट कर सकते हैं, कैसे और नए प्रोजेक्ट्स कर सकते हैं, इनसाइट में जाके थोड़ा समझना. तो इसलिए जो है वो, और वैसे भी ज्ञान का जो ओवरऑल प्रोग्राम है जो वारसी सर की तरफ से ये कोऑर्डिनेट होता है. इससे पहले भी हमने मेडिकल कॉलेज में 2-3 ज्ञान के कोर्स जो है वो अटेंड किए हैं, कंप्लीट किए हैं अंडर जो है वो कोऑर्डिनेशन ऑफ प्रोफेसर तमकीन रब्बानी, प्रोफेसर सीमा हाकिम मैम के. चूँकि वो हमारी फील्ड से था तो वो हमारे लिए खासा जो है वो बेनिफिशिएंट था. ये थोड़ा सा फील्ड से हटके था, लेकिन हमें इस फील्ड के साथ कोलैबोरेट करके अपनी मेडिसिन को और फ्लरिश करने का की ख्वाहिश थी. और चूँकि अपनी रूटीन में डेली में तो हम इस फील्ड में आ नहीं सकते या बहुत ज़्यादा इन डेप्थ जो है वो पढ़ नहीं सकते, गेन नहीं कर सकते. इसलिए ये था कि कम से कम इस 5 दिन में हम बाउंडेड रहेंगे और कुछ ना कुछ तो ज़ीरो से कुछ ना कुछ तो अपलिफ्ट होंगे. हालांकि बहुत सारी चीज़ें बाउंस कर गई, जहन से ऊपर से निकल गई, लेकिन बहुत ही अच्छा लेक्चर था. और जो खासकर जो फॉरेन फैकल्टी सर ने बुलाई थी, हम तो उनसे बहुत इम्प्रेस हुए. उनकी एज, लेकिन उनका स्टेमिना, उनका जो नॉलेज तो था ही, खैर वो है ही स्टीम्ड फैकल्टी बाहर के, लेकिन उस एज में वो स्टेमिना, हम खासा इम्प्रेस हुए सर के लेक्चर और सर के ट्यूटोरियल से. और बहुत ही इन डेप्थ में उन्होंने जो ड्रग के मतलब एक नैनो मॉलिक्यूल्स और उस लेवल पे उन्होंने बात की. अपना जो एक्सपर्टीज़ थी उन्होंने अपना एक्सपीरियंस शेयर किया. बहुत सारी जो हम लोग को जो है वो इंथुजियाज्म दिया एक तरह से कि आप किस तरह अपनी फील्ड में रह के और किस तरह से उसमें ड्रग डिस्कवरी या डिलीवरी से रिलेटेड पार्ट पे आप कैसे काम कर सकते हैं. साथ में वो इंडस्ट्रियलिस्ट जो थे वो सर ज़ियाउद्दीन सर, खासा इम्प्रेसिव लेक्चर, बहुत ही अच्छा था सर. थैंक यू सो मच सर. शायद अभी है नहीं यहाँ पे. हाँ, बहुत ही अच्छा, और बहुत अच्छा उनका प्रैक्टिकल पर्सपेक्टिव था कि किस तरह इंडस्ट्री का जो है वो क्या स्ट्रेंथ है ड्रग डिस्कवरी से रिलेटेड, और फिर उसको मार्केटिंग करना, फिर उसको डिलीवर करना, और क्या चैलेंजेस इस वक्त हम फेस कर रहे हैं. उन्होंने उस पर खासा जो है फोकस किया. और हम किस तरह से आज का जो मॉर्निंग सेशन था उस पर कि आप जो कर रहे हैं उसको कैसे पब्लिक डोमेन में लाएं, और ज़रूरी क्या है, ज़रूरत है उसकी कि पब्लिक डोमेन तक बात आए, वरना आप जो करते हैं वो वहीं का वहीं सबसाइड होगा. कोलैबोरेशन पे उन्होंने बहुत एम्फेसिस दिया. तो बहुत सारी चीज़ें नहीं पता चली. जो थोड़ी सी कमी सी लगी वो भी बताना ज़रूरी है, क्योंकि फीडबैक दोनों होता है. खासा सब कुछ बहुत अच्छा था. और जो कमी हमें लगा कि सर जो पूरा प्रोग्राम था ऑल ओवर, हमने फीडबैक में भी इस चीज़ को मेंशन किया है, वो कहीं ना कहीं क्लिनिकल पर्सपेक्टिव से थोड़ा सा जोड़ना था. 1-2 सेशन फिजिशियन, क्लिनिशियन, पैथोलॉजिस्ट, मीन्स क्लिनिकल फील्ड से उनकी जो एक्सपर्टीज़ हैं, और वो कहाँ से ड्रग जो डिस्कवरी हो रही, देन डिलीवरी वाला पार्ट जो है वो कहाँ से पैथोलॉजी से कब काउंटर होगा या कहाँ से अटैच होगा, कैसे हम यहाँ पे जैसे स्ट्राइक करें इस प्रॉब्लम में, एंटीबायोटिक दे रहे हैं फॉर पर्टिकुलर जो भी प्रॉब्लम्स है उससे रिलेटेड, तो ये मॉलिक्यूल यहाँ पे करता है, इस तरह से पार्ट करता है. तो वो थोड़ा हमारे लिए इंटरेस्टिंग होता. तो आगे के लिए रिक्वेस्ट है, इन फ्यूचर कभी इस टॉपिक पे अगर प्रोग्राम करें तो सर प्लीज़, चाहे आप इंडियन फैकल्टी ही रखें, लेकिन क्लिनिशियन पर्सपेक्टिव पे क्लिनिशियन का 1-2 ट्यूटोरियल लेक्चर एक्सपेरिमेंट हो तो बहुत ही अच्छा है. और बस लास्टली कहना चाहेंगे कि सर की हॉस्पिटैलिटी बहुत ही अच्छी थी. ग्रेट हॉस्पिटैलिटी, वी एंजॉय द 5 days. बहुत ही अच्छा था. और ऑल ओवर बहुत अच्छा लगा हमें. जो है वो हम मेडिसिन फील्ड से निकल के इंजीनियरिंग फैकल्टी और नैनो टेक्नोलॉजी में आए, तो थोड़ा अपनी ही यूनिवर्सिटी में ऐसा लगा कि हम नए एक जोन में आए. तो थैंक यू सो मच सर. थैंक यू.
Thank you for the nice feedback, and I love these kind of feedback which also have some critical components जिससे हम कुछ सीख सकें. So we'll definitely take it, and we hope to improve the course in future. से ये मल्टीडिसिप्लिनैरिटी से ही शुरुआत हुई थी बात की, मल्टीडिसिप्लिनैरिटी पे ही ख़त्म कर रहा हूँ कि yes, this is also up to the clinicians to come forward and take the lead and involve in the discussions that would have made the course much more richer. तो क्लिनिशियंस आगे आए, दे हैव गॉट अ लॉट ऑफ रिस्पॉन्सिबिलिटी, उनके पास क्लिनिकल ड्यूटीज़ हैं, लेकिन उसी में से वक्त निकाल करके आगे आएँ और इसको एनरिच करें कोर्सेज को विद डिस्कशंस, विद lot of things that they can add to this. Right, so if anybody else has nothing to say, I would like to request Professor Varsi for giving us his remarks and insights, because he has seen a lot of courses happening all over India, and his insights are very necessary. Sir.
Dr. Azhar Aziz Saab, all the participants, colleagues, students, a very good afternoon to all of you, and Assalamu alaikum. Actually, I will start with a quote which says, like, it is peculiarity of the knowledge that those who really thirst for it always get it. And this is every step is a forwarding a step, like towards success, towards and reaching the gap, towards reaching to the goal. So it's not in a complete, like one GIAN course is not incomplete itself. So we are moving forward, learning something, moving towards success, moving towards filling the research gaps, learning something new. And this world is not for core subject. This time we are moving, the world is moving towards interdisciplinary and multidisciplinary approach. And I'm good that people from different backgrounds here and in large numbers are here. And so this is one step. The condition for this course is like we have to have a foreign expert to give you an exposure, what kind of research in foreign countries are happening, so to have an insightful. So as I said, it's not to have everything to understand, to get, but it is one step towards getting ourselves enriched academically, scientifically. So this is good things that is happening, initiated by the Government of India, Ministry of Education. And I'm sorry I missed the entire program. I was away from the country, just came this morning, and I promised Azhar Sir that I will be here at least for the validity to interact with you all. The good news with the GIAN is that this year, with a maximum limit of 40, we were able to somehow submitted 42 proposals to the GIAN for the next phase. So this is the second last of the phase 4th of the GIAN program. One more to go by the same Professor Dr. Azhar Saab. It's going to start from 15th of December, I think so, right now. So this is good exposure for the people. When I go outside, we see, like, we learn different perspective. As she rightly said, from the Tibia background, like Unani background, you have to come to the nano part, then in engineering part, like, you know. So this world is not limited to one domain. It has become multi-domain specialized world. So we have to be as much as possible in a multilingual scenario. We will be successful, more successful in our life. So with these words, I would like to thank and congratulate Dr. Azhar Saab, and congratulations to all of you for a bright future, and looking forward to see you and some other GIAN program in near future. Thank you very much. Thank you very much.
Thank you, Professor Varsi Saab. And I just want to mention one thing here. We are really nano. Nano in the team. You know how many people were there with us to organize this entire 5-day event, getting a foreign faculty from abroad? We were just a couple of people exactly. So we were nano, but nano makes a big difference. So we try to do our best. And I would like to thank all the organizers, especially Dr. Kasid Anwar, Dr. Sayyid Afzal Ahmed, and my PhD student, Sayyida Shabi Zafar. And definitely, in absentia, I'm saying thanks to all the university functionaries, administration, Honorable Vice Chancellor, Registrar, and there are a whole big list there. So, and thanks to the funders, the Ministry of Education. They have given us the funds to do all this activity. And last but not the least, you are the focus, the participants. I'm so thankful to all of you for your continued presence, for your involvement, for asking questions, getting participation, your enthusiasm, because that is what is the denominator for success for any course, for any teaching or academic activity. Thank you so much. So I would like to request Professor Varshi to hand a memento to the organizer. Dr. Sayyid Afzal Ahmed is here. Unfortunately, another organizer, Dr. Salia Naseem, she broke her leg. So she had undergone a big surgery, and that's why she's not here. But she was managing things remotely, and thanks to her as well. And now I would request Professor Varsi to take his valuable time in giving away these certificates. We'll call them one by one. And I think that will be easier to, so that Adil Muhammad, Mariam Siddiqui, Farheen Dayas, Mohsina Ahmed, Fatija, Mohsina Ahmed, Fatija, Muzammil Liaquat Mir, Mohammed Aqib, Mohammed Aqib, Anup Verma, Anup Verma, Isra Aman Aziz, Dr. Tahmida Zinat, There'll be a group photograph after this activity, so please stay for a few more minutes if you can.
Asma Jigan, Nausheen, Malik Rehan Ahmed, Malik Rahman, Malik Rehan, Sheikh Anand Sarfraz, Meena Miraj, Abdullah Manan, Abdullah Manan, Saiful Zafar, Saiful Zafar, Muhammad Salim, Mehak Tabi, Sofia Rahim, Sofia, Dr. Sameema Khatun, Shirjeel Ahmed Siddiqui, Anam, Anam, Muhammad Zulfikran, Muhammad Zulfikran,
Muhammad Zulfiklan. Zulfiklan. Exotic name, huh?
Muhammad Shahid, Muhammad Shahid, Afshan Anam, Arfin Hussain, Sabah Parveen, Muhammad Shoaib Khan, Dr. Sheikh Shahbaz, Maharat Siddiqui, Maharat,
Thank you so much, everybody. I just would like to say a final thank you, but it will not be complete if I don't mention Zainab Zahid, Suzanne, Azam, and Abdurrahman, who have been working tirelessly behind the scenes without getting even acknowledged. Thank you once again. If you can come on the stage, we'll have a photograph and then we are all good to go. आप थोड़ा सा अरेंज करवा देंगे
आप यहाँ आ जाइए
जी जी मैं
टिकट लेना है बस यहाँ आ जाए
नीचे स्टेज पे भी आ जाए आप लोग
कुछ नीचे भी आ
and you can come on the stage